Hey, hey, Anna, hey, not thousand. বন্ধ দিয়েছি দরকার প্লেট মার দেবো এলাম না এটা ছুঁই দেবো ছুঁই দেবো তো দেখবি কই লাগলে করবি না সহ্য রিচার্জ দিয়ে দে মানে কর Hello.
Yes, yes, we will
Yes,
start.
sir. Can start, sir?
And wait, madam, wait, Siraj sir is not now here. I think we'll join. So we'll
Okay,
wait
sir.
1 2 minutes.
Okay, sir. Raji?
Yes, ma'am, I'm here, ma'am. Hello, ma'am. Am I audible, ma'am?
Yes, ma. Yes, ma. You can start.
Yes, ma'am. Yes, ma'am. Shall I start, ma'am?
Yes, you can start.
Yes, ma'am. A pleasant evening to one and all present here. I am Rajrajeshwari, Research Scholar of Botany Department, Avinash Lingam Institute for Home Science and Higher Education for Women, Coimbatore. I take immense pleasure in performing embassy for this event. Rise up, start fresh, see the bright opportunities in every day. I wholeheartedly welcome all the dignitaries, research scholars, and faculty members of various departments. One song can change a moment, one idea can change a world, one step can start a journey, but one prayer can change even the impossible. I request Yangshan Dolmakom, Avnashlingam Institute for Home Science and Higher Education for Women, Coimbatore, to sing the prayer song. Sangachatvam Sangadhatvam Sambho Manasi Janata Devavagamyate
Meghna?
Yes.
Yes,
Thank
yes.
you. It was wonderful. Now, I would like to call upon Dr. Pampi Ghosh, Assistant Professor and Director, Department of Botany and Secretary of the Seva Bharati Mahavidyalaya, to present the welcome address.
So, good evening to all the respected guests, resource persons, students, researchers, participants, and ICT personnel. This is our second day of this conference, and we are gathering here for make it complete. Our conference is on biodiversity building and natural resource management. This is our second day, and without hampering time, I welcome all the resource persons and all the personnels and all the guests again. Here, three institutions are actually jointly organizing this conference. Two from West Bengal, my institution, that is our institution, Seva Bharati Mahavidyalaya, Kapgari Jhargram from West Bengal, and another GGDC, Lalgor, Jhargram, and another Avinash Lingam institution, that is a university, Avinash Lingam Institute for Home Science and Higher Education for Women, that is deemed to be university. Here three departments, namely Zoology, Botany, and Department of Biochemistry and Biotechnology and Bioinformatics are here. So welcome all again, and please proceed for next.
Thank you, ma'am. Up next, I would like to call upon Dr. Debaprasad Sahu, Principal, Seva Bharati Mahavidyalaya, to present the greetings.
He is not here.
Okay,
He is
ma'am.
not here, so proceed.
Okay, ma'am. Now, I would like to call upon Professor J.P. Keshri, Department of Botany, University of Burdwan, West Bengal, to present the greetings.
Okay, good evening, everybody. In this forum where all are living in our office, our homes, and still we are connected with each other. So, uh, this is welcome greetings from my behalf because, as all of you know, that India is a biodiversity-rich country, and unfortunately, uh, we have not realized properly the importance of biodiversity in our country. So this initiative is a very grand initiative because capacity building is also important. I can cite one few examples. One of my colleagues, Professor Sen, he is at North Bengal University. He told that his teacher from Japan, he came to uh, his North Bengal University and visited a few places of uh, the Darjeeling Hills. So, what he was doing, he was just moving from one place to another and then again coming back. So, the professor Sen asked, so what are you doing, sir? But actually, I wonder with this biodiversity because in a very small place I am getting 50 or 100 plants, but in our place in many parts of the temperate countries, such biodiversity is not. So biodiversity is our resource, and the knowledge of biodiversity, identification of plants, how they are utilized, that is important. We may do molecular biology, we may do biotechnology, but everything is connected with that. So our knowledge of the biodiversity is very important. So I shall congratulate uh, for this forum where we are here to convey our thoughts and interact with each other. So thank you to all of you uh, for this opportunity to interact with you. So thank you very much.
Thank you, sir. Up next, I would like to welcome Professor Siraj Datta, Controller of Examinations, Vidyasagar University, Midnapur, West Bengal, to present the greetings. Sir, are you there, sir?
So please unmute your mic, sir.
Professor Siraj Datta, sir, are you there?
Sir is there, ma?
Hello, am I
Yes.
audible?
Yes, sir.
Thank you to the organizer to invite me such an auspicious event. And due to my, because all of you know that the university exams just knocked the door from January onwards, 5th sem, 3rd sem, and first sem examinations are coming. That's why I'm unable to join this though my background is of biology and biotechnology, so I take interest to when I'm invited to join this forum actively, and but I'm unable to view this session or actively joining the session. But wish you all the best for these 2 days, and hope um, all the things which are going on in these 2 days are very resourceful and meaningful for our students as well as the teachers and all the other stakeholders who are directly involved in these 2 days um, workshop or seminars. Thank you.
Thank you, sir. Now, I would like to welcome Dr. V. Gayatri, Assistant Professor, Department of Botany, Avinash Lingam Institute, to shed a little more light upon today's events.
Thank you, Meghna. A warm good evening to one and all present here. The introduction of many minds into many fields of learning along a broad spectrum keeps alive questions about the accessibility, if not the unity of knowledge. This was by Edward Levy. So with this quote, I would like to introduce the resource persons of today's session. In day 2 of the National E-Conference, we have Dr. R.K. Mishra as the first speaker, whose talk is on assessment of biophysical parameters and plant diversity of similipar, an ecological approach in conservation planning. He is Assistant Professor, Department of Wildlife and Biodiversity Conservation, Maharaja Sri Ram Chandra Banjar Dio University, North Orissa. The second speaker of the day is Professor Vidina Jha, retired principal and Professor of Botany, Ellen Mithila University, Bihar, on the topic utilization of components of biodiversity in floodplains of Mithila, North Bihar. The next speaker on the second day of the conference is Dr. Nirmala Gogai, Assistant Professor, Department of Environmental Science, Tezpur University, Assam, on the topic biodiversity under changing climate, mitigation of threats. The last inviter lecture of this 2-day conference will be given by Dr. D. Thangamani, Scientist, FGRM Division, IFGTB, Coimbatore, on the topic conservation and characterization of forest genetic resources and its utilization for our future. So the talk by resource persons will be followed by technical session 2 and technical session 3, in which paper presentation will be done by scientists, research scholars and staff members in various areas of research. The rapporteur for the technical session 2 will be Dr. Pampi Ghosh, HOD, Department of Botany, Seva Bharati Mahavidyalaya, Kapgari, and for technical session 3, Dr. G. Maheshwari, Assistant Professor, SS, Department of Zoology, Avinash Lingam Institute for Home Science and Higher Education for Women, Coimbatore. Finally, the reporting of day 2 will be done by Dr. Sri D. Mitra, Assistant Professor, Department of English, Government General Degree College, Lalgarh, and Dr. S. Sudha Devi, Assistant Professor, Department of Biochemistry, Biotechnology and Bioinformatics, Avinash Lingam Institute for Home Science and Higher Education for Women, Coimbatore. Finally, the vote of thanks will be delivered by Dr. V. Gayatri, Assistant Professor, Department of Botany, Avinash Lingam Institute for Home Science and Higher Education for Women, Coimbatore. Thank you for giving me an opportunity to do the introduction for the day 2 of the national conference. Thank you.
Thank you, ma'am. Now, I would like to welcome our first resource person for today, Dr. R.K. Mishra, Assistant Professor, Department of Wildlife and Biodiversity Conservation, MSCBD University, Takhtpur, Baripada, Odisha. On to you, sir.
Okay. Thank you, madam. So, I am audible? Hello?
Yes, sir.
Yes, sir.
Am I audible? Okay. Share karo slide. Hello? Hello?
Yes, sir. Yes, sir.
The sharing is, the slide
Yes,
is
sir. Sharing is visible, sir. If
Okay,
possible, can change it to slideshow mode, sir.
Okay, madam. So, good evening to everybody. And first of all, I am very much thankful to the organizers. I am very much thankful to the organizers and Dr. Baidyanath Jha, LN Mithila University, Dr. Gayatri, Dr. Das, Dr. Pampi Ghosh, Sabha Bharati Mahavidyalaya, Siraj Kumar Datta, Registrar, Vidyasagar University, Dr. Gayatri, Department of Botany, Avinam Lingam Institute, Coimbatore. So today, I am very much pleased to the organizers to select me as one of the resource person of this national seminar on biodiversity conservation, and from where I start my journey in research, that is in Similpal Biosphere Reserve. And today I elaborate my topic entitled Assessment of Biophysical Parameters and Plant Diversity of Similpal and Ecological Approach in Conservation Planning. So first of all, I want to tell you that why we can monitor and assess the biological parameters, or why we can go for the monitoring and assessment of the different elements of biodiversity. For the assessment of different elements of biodiversity, at that time, the forest covers of different parts in the world as well as also in our country came into limelight. So when it is came into limelight, and why it is came into limelight, because before 1986, there are three basic elements for different types of living organisms, mostly the human beings, that is known as the food, shelter and clothing. But now, after the establishment of EPA Act 1986, the basic elements are changing from food, shelter and clothing to air, water and soil. And without air, we never survive. Without water, our protoplasmic activity or the protoplasmic activity of the cell is not properly functioning. And without soil, the plant cannot survive, or the plant cannot survive or perform the different types of activities and its growth and development. As a result of which, we can go for the conservation of the most important biological element, that is known as the plant. So in this context, I want to tell you that forest is a good servant but a bad manager. And why forest is a good servant but a bad manager, I can start the lecturer from this point, that is forest ecosystem of Similpal can provide different types of activities to the different types of living organisms as well as also to the human beings. So the forest ecosystem of Similpal is responsible for the water conservation, climate regulation, nutrient cycling, soil protection, and due to soil protection, there is the prevent of water and soil erosion. And due to the forest ecosystem of Similpal, the plant litters and animal litters are deposited on the forest floor, and due to add of litter, that increase the soil humus. And when there is the increase of soil humus, ultimately that soil humus is leached into the soil or into the deeper part of the soil, and the roots of the plant can absorb the nutrients, and that is useful for the better growth and the development of the plants. In another hand, how the plants are useful for us, the plants are absorbing or taking carbon dioxide, or that is known as the carbon dioxide fixation for the photosynthetic activity, and due to after the photosynthetic activity, one of the gas is released into the outer atmosphere, that is known as the oxygen. And without the photosynthetic activity, if no oxygen is released into the outer atmosphere, then the different types of living organisms are facing problem in their inhalation activity. And the forest ecosystem is also acting as the pollutants decomposition, also acting as the windbreak, and the forest ecosystems of Similpal are the different types of plant life forms of the Similpal is responsible for photosynthetic activity, and as a result of which the different plants are providing food material to the different types of animals, as a result of which that is the increase in organism production. And ultimately, though the forest ecosystem is the habitat for the different types of animals, the forest ecosystem also responsible for to conserve the different types of wild animals, birds, and plants. And ultimately, the forest ecosystem also reduce the disease as well as also the pests. So when we concerned about the forest ecosystem of Similpal, Similpal is one of the biosphere reserve of the state of Odisha, and it is the 9th biosphere reserve which was recognized by the UNESCO. Similpal forests are located in the district of Mayurbhanj at 21 degree 16 minute to 22 degree 08 minute north latitude, and 86 degree 4 minute to 86 degree 37 minute east longitude. The highest peak of Similpal is Meghasani, and the height is 1165.5 meter, and it was declared as the Biosphere Reserve in June 1994. The Similpal Biosphere Reserve is one of the hotspots of biodiversity in the state of Odisha because it encompasses or it comprises of around 1100 plant species, and in the Biosphere Reserve there are 5 different types of forest covers. Most dominant ones or the most dominant types of forest covers found in Similpal Biosphere Reserve are tropical moist deciduous forest, dry deciduous forest, dry evergreen forest, and the dry semi-evergreen forest. So this is the evergreen forest patch of Similpal Biosphere Reserve. This is the moist deciduous forest patch of Similpal Biosphere Reserve, and this is the dry deciduous forest, and this is the semi-evergreen forest. So not only the Similpal is the containing not only the Similpal, there is the presence of different types of forest covers, but also the different types of flora and fauna, as a result of which Similpal is considered as one of the hotspots, and it is also one of the biosphere reserve and included under the UNESCO program or under the UNESCO list of biosphere reserve. These are the different floral components of Similpal Biosphere Reserve. This is Rauwolfia serpentina, this is the Carya arborea, and this is Solanum virginanum, and this is Cleistanthus colinus, and these are Dendrobium transparens, Nitamula, and this is one of the endangered plant species, Nitamula, and the Curcuma species, Bahunia variegata, Campharia rotunda, Area megasensis, Butia monosperma, Caliandra hematocephala, and Anosis latifolia, Caparis gelanica, Cassia didymo, Bacteria, Cassia hitsuta. So likewise, the different types of that is Sterculia villosa, Cochlospermum religiosum, Dicrostachys cinera, Indigofera cassioides, Curculigo arcadis, Pterospermum xylocarpum, Gloriosa superba, Lepicanthes tetraphylla, Syzygium aromaticum, Clerodendrum ceratum, Lobelianaco, Tinifolia, Solanum macrophyllum. Likewise, these are the different types of lichens are also extracted or also observed from the Similpal Biosphere Reserve. So it is one of the hotspot or containing nearly about 1100 plant species as well as also many number of animal species. In many number of animal species, there is nearly about 342 species of birds, that is 50 species of mammals, and nearly about 59 species of amphibians, and all other small and large animals also inhabited in the Similpal Biosphere Reserve due to the good coverage of vegetation cover. Now the question arises, due to the progress of time, due to the spatial change, and another one is known as the temporal change. What happens? The different types of anthropogenic pressure is imposed on the forest covers of Similpal Biosphere Reserve, as a result of which the anthropogenic activity is increased day by day on the forest covers of Similpal. So our first aim is how to quantify the disturbance that is imposed upon the forest covers. Not only we can tell it that such amount of pressure is imposed on the biosphere reserve, we must have to calculate it, or we must have to quantify it. So how we can quantify it? So for the first time, we can calculate one of the index to know about the disturbance, that is known as the disturbance index. The disturbance index in case of Similpal Biosphere Reserve, in terms of percentage, that is all the Similpal Biosphere Reserve, which area is 5,569 square kilometer, and that area is divided into 3 different zones. And what are the 3 different zones? One is known as the northern sector, one is known as the southern sector, another one is known as the central sector. And in all the 3 sectors, we have selected 18 different study sites. And out of the 18 different study sites, some of the study sites are disturbed, some are moderately disturbed, and some are the undisturbed sites. So how we can categorize the area into disturbed site, moderately disturbed site, and undisturbed site? We can categorize the area into 3 different groups, that is the disturbed one, moderately disturbed one, and the undisturbed one, depending upon the disturbance index. When the disturbance index in terms of percentage is greater than 30, then that is called as the disturbed site, and when it is 15 to 30, then that is considered as the moderately disturbed, and when it is less than 10, that is considered as undisturbed sites. So in all the three different sectors, all the three different sites, that is the disturbed site, moderately disturbed site, and the undisturbed site. So all other biophysical parameters are also differ from each other. Biophysical parameters, some of the biological parameters, as well as also some of the physical parameters. The physical parameters like the edaphic ones, as well as also the light intensity, and that too are considered as the physical parameters, and the biological parameters like the density of the plant, that is the crown area of the plants, or the basal area of the plants are considered as the biological parameters. So here, in case of the physical parameters, the soil moisture content is increased from the disturbed site to the undisturbed site. The pH value is less acidic in case of a disturbed site and more acidic in case of undisturbed site, and through moderately disturbed sites. So organic carbon content in terms of percentage increased from the disturbed one to the undisturbed one. Nitrate nitrogen content, total nitrogen content also increased from the disturbed to the undisturbed one. Available phosphorus is also increased from the disturbed to undisturbed one. Available potassium, but available potassium is showing the negative trend, that is decreasing from the disturbed one to the undisturbed one. The tree density is increased from the disturbed to the undisturbed one. Shrub density is decreasing. Herb density is also decreasing from the undisturbed one to the undisturbed one. The tree crown area is 23.16%, and in moderately disturbed that is 31.42, and in undisturbed that is 45.42. What it indicates? The tree crown area indicates that if the crown area is more than 40%, then that is called as the closed canopy. And if it is less than 30%, then that is called as the open canopy. And the intermediate range is observed in case of the moderately disturbed sites. When we consider about the light intensity, that is photosynthetically active radiation, which was measured in terms of micromole per meter square per second, the light intensity on the forest floor is more in the disturbed site and less in the undisturbed site, because in undisturbed site, due to high canopy area, the light penetration on the forest floor is decreasing. But in the disturbed one, the light penetration on the forest floor is more due to the very less canopy coverage. Now we can come to how we can calculate the disturbance index, and another one is also there, that is known as the Ramakrishnan index of stand quality. So from these two different diversity indices, we can know about the quality of the study sites or the quality of the forest stand. So first one is the study of Ramakrishnan index of stand quality. The Ramakrishnan index of stand quality was first measured by Professor P.S. Ramakrishnan, who was the professor of environmental science in Jawaharlal Nehru University, New Delhi. So risk is equal to summation Ni into pioneer index divided by N. And what is the Ni and what is N? Ni and N is very much similar to the Shannon index, or that is known as the species diversity index. Ni indicates the number of individual species, and the N indicates the total number of species present in one site. And how we can calculate the pioneer index? the pioneer index is divided into, or the pioneer index, we can provide 3 values to the pioneer index. And what are those 3 values? First value is known as pioneer index is 1, for which species, or for which species pioneer index is 1 for the species whose seedlings can establish in the closed canopy area but need small canopy gaps to grow up. Such type of species are considered, or such type of species are provided the value 1 as the pioneer index. Similarly, pioneer index is 2 for the species whose seedlings establish in small gaps but need small to medium sized gaps to grow up. And the pioneer index is 3 for the species whose seedlings need large canopy gaps for both establishment and growth. So by taking these 3 different pioneer index values, we can put the pioneer index value here, and the number of individual species Ni, and N is known as the total number of species in the site. Then we can calculate the risk, that is known as Ramakrishnan index of stand quality. The Ramakrishnan index of stand quality value is a maximum up to 4, not beyond 4. So then another index was also calculated to know about the disturbance of the study sites, that is known as the disturbance index, and that index was calculated by us, by our team, for the first time in Similpal Biosphere Reserve. That is known as the disturbance index in terms of percentage. How we can calculate the disturbance index in terms of percentage? That is the total basal area of damaged plants. Which plants are considered as the damaged plants? The plants which are partly damaged, or the plants which are standing dead, the plants which are dead fallen, or some parts of the plant is damaged either due to the anthropogenic activity or due to any type of animal intervention. So that is considered as the damaged plants. Then that is divided by the total basal area of living plants. Living means that is known as the undamaged plants and the damaged plants into 100. Then we can calculate the disturbance index. So based upon these 2 diversity indices, that is known as Ramakrishnan index of stand quality and the disturbance index, we can divide all the 18 study sites what I am already presented in the that is in the map of the Similpal Biosphere Reserve. These are the 18 different study sites that is located in different directions and different altitudes of the Similpal Biosphere Reserve, because this is known as the core zone of the Biosphere Reserve, and that surrounds the buffer zone, and again, and the periphery one is known as the transition zone. In all the 3 zones, the 18 study sites were selected, and their disturbance index and Ramakrishnan index of stand quality was calculated. So, so this is about all the 18 different study sites. These study sites, site 1 to site 5, that is study site 1 to 5 and 8 and 9. These 7 study sites are located in the transitional area, and that is considered as the disturbed sites. And site 6, 7, 10 and 12, these 4 sites that are located in the buffer zone, and another 7 sites are located in the core zone of the Biosphere Reserve. So in all the disturbed sites, you have to find out the basal area in terms of meter square per hectare, and what is the basal area of the living plants, that is undamaged plants, and this is the basal area of the damaged plants. And this is the total basal area, and the basal area of damaged plants divided by total basal area of the undamaged and damaged plants into 100 is considered as the disturbance index, and that is DI percentage. And from this table, it indicates that in all the disturbed sites, the disturbance index is greater than 30. And in the sites which are located in the buffer zone, that is the disturbance index varies from 15 to 30, and that is considered as the moderately disturbed sites. And in all the 7 sites located in the core zone, the disturbance index is less than 10%. That's why that is considered as the undisturbed sites. Similarly, in association with the disturbance index in terms of percentage, when we can calculate the Ramakrishnan index of stand quality, in case of the disturbed ones, the Ramakrishnan index of stand quality is more in comparison to the sites located in the buffer zone and the sites located in the core zone. In the core zone, the Ramakrishnan index of stand quality value is always less than 2. And in case of disturbed sites, Ramakrishnan index of stand quality value is nearly approaching 3. So based upon these 2 diversity indices, we can divide the 18 different study sites into the 3 different groups, that is known as the disturbed ones, the moderately disturbed ones, and the undisturbed ones. So in case of disturbed sites, such type of anthropogenic activities are going on, that is the logging practice, and the forest is converted into agricultural fields, and how the forest cover loss is taking place due to different types of anthropogenic activities. Not only these are the anthropogenic activities of imposed on Similipal Biosphere Reserve, but also some other anthropogenic activities are also going on inside Similipal Biosphere Reserve, that is known as the forest fire, the grazing of the different types of domestic animals, and the plucking of leaves by the tribal population of Similipal Biosphere Reserve for their livelihood. And this is also conversion of the forest cover into agricultural fields due to cutting down of or lopping of different types of plant species. Now we can come to another type of biophysical parameter that is known as the photosynthetically active radiation, and how much amount of light is available on the forest floor at different time periods of a day, from 7 a.m. up to 5 p.m. and what is the photosynthetically active radiation on the forest floor in 3 different seasons, that is during summer, during winter, and during the rainy season. And the peak is marked at 12 noon to 1 p.m. in every season, and nearly what it indicates, this is in the disturbed condition, and this is the moderately disturbed condition, and this is the undisturbed condition. In 3 different conditions, in 3 different seasons, this is the amount of photosynthetically active radiation on the forest floor. And as per the study of Kolari et al. 2006 and Singh and Chaturvedi et al. 2000 and Singh and Chaturvedi 2017, how much amount of photosynthetically active radiation is there in different tropical forest covers of India, that is nearly about 40 to 400 micromole per meter square per second. And here, in case of Similipal Biosphere Reserve, that is within the permissible limit, as a result of which the forest cover, mostly in the undisturbed and moderately disturbed sites, that is in good condition. And in case of disturbed site, that is approaching towards the peak. So next parameter, the next physical parameter is the temperature. So temperature value was also measured at different time periods of a day, starting from the 7 a.m. to 5 p.m. in 3 different seasons, in 3 different disturbance conditions. And in case of the forest covers, the temperature, which one is the favorable temperature? The favorable temperature is 28 to 29 degrees centigrade, and that may up to 31 to 32 degrees centigrade. And that leads to the increase in the gross primary productivity of the different varieties of the plants or the life forms. Either that is the herb, that is the shrub, that is the tree, or that is the climber. For the different layers of the vegetation cover, the amount of temperature required, or which one, or what is the favorable temperature for the different vegetation layers, that is nearly about 28 to 29 degrees centigrade, or that may increase up to 31 to 32 degrees centigrade, and that is responsible for the good gross primary photosynthetic, gross primary productivity of different vegetation covers. And here what we have marked, the temperature regime of Similipal Biosphere Reserve is also within the permissible limit. The next physical parameter is known as the relative humidity. So why we can measure the relative humidity? Because the relative humidity is mostly related to the moisture content, and also it is also correlated to the moisture content of the soil. In relative humidity, due to very high relative humidity in 3 different disturbance regimes of the Similipal Biosphere Reserve, or in 3 different disturbance gradients of the Similipal Biosphere Reserve, in Similipal there is the greater visibility and high diversity of the orchid plants. The Mishra, one of the personnel from Odisha, that is Sharath Mishra, already documented 94 species of orchids from Similipal Biosphere Reserve, and out of these, one of the orchid is the endemic one. And why Similipal Biosphere Reserve is very high population of orchid or high diversity of orchids? Only due to the high humidity value or the relative humidity value. So now we can come to another physical parameter, that is known as the soil. So when we can consider about the soil, in case of the sand, in case of the silt, what we have observed, in case of sand and silt, we have observed that the sand percentage is decreasing from the disturbed site to the undisturbed site, and not only from the different disturbance gradients, but also along with the soil depth. Along with the soil depth, the sand percentage is also decreasing. And similarly, in case of silt, the percentage of silt content is decreasing from the disturbed one to the undisturbed one, and that is also decreased with the different depths of soil, that is 0.10 to 20 to 30 centimeter soil depth. The silt percentage is also decreasing. But the reverse trend was marked in case of clay percentage. In case of clay percentage, the clay percentage is increased from disturbed site to the undisturbed sites, as well as also the clay percentage is increased from the surface layer, that is 0.10 centimeter depth to 20 to 30 centimeter depth. And that indicates the good retention capacity of water in the soil of Similipal Biosphere Reserve. And in case of a bulk density, the bulk density is decreasing from the disturbed condition to the undisturbed condition, and the bulk density is increasing with respect to the depth. But in case of porosity, the soil porosity and soil moisture content, both the parameters, which are the two indicators for the growth and the development of the plants, are also these two parameters of the soil, physical parameters of the soil, also indicating about the diversity potential of the different groups of plants in case of Similipal Biosphere Reserve. The porosity and soil moisture content is increased from disturbed one to the undisturbed one, and also the soil porosity and soil moisture is increasing from the along with the different depths or across different depths. That is from 0.10 centimeter. Sorry, I'm putting the wrong thing, telling the wrong thing, because the data indicates that the along with the depth that is decreasing, and with the across the disturbance gradient that is increasing, the porosity and soil moisture content. So the question arises, why the porosity percentage and the soil moisture percentage are increasing from the disturbed one to the undisturbed sites, and both the parameters, that is porosity and the soil moisture content, is decreasing with the increase in depth. The reason is that, so on the top soil or on the surface, there is accumulation of more litter, and when there is accumulation of more litter, and due to microbial activity or due to very favorable climatic condition, the microbial population is more, and that can responsible for the degradation of the different types of the litter on the deposited on the forest floor, as a result of which the soil organic carbon content is increased. And when soil organic carbon content is increased and the soil humus content is increased, that leads to maximum porousness of the soil, as a result of which across the disturbance gradient from the disturbed one to the undisturbed one, the porosity percentage is increased, as well as also soil moisture content is increased. So now we can come to the pH, the different chemical parameters of the soil. So, so when we can concerned about the different chemical parameters of the soil, like the physical parameters, the chemical parameters also influence the different types of the vegetation covers of Similipal Biosphere Reserve. How it can influence or how it can regulate the different types of plant life forms of Similipal Biosphere Reserve. So when we concerned about the pH value is less acidic in case of disturbed sites and more acidic in case of undisturbed sites. And why more acidic and here why less acidic? Because due to more density of the plants, more canopy coverage, more amount of litter is deposited on the forest floor, and more amount of humus is there. And when more amount of humus, more amount of organic carbon is on the forest floor or on the soil, that leads to the acidic pH. And here in case of organic carbon, the organic carbon content, the nitrate nitrogen content, as well as also the total nitrogen content. All these three variables, all these three chemical parameters of the soil are increasing along the or across the disturbance gradient. That is increasing from disturbed sites to undisturbed sites. And the reason is, that is the litter as well as also the rate of decomposition. So we are also publishing 2 to 3 papers in very good journals, that is forest ecology and management, and about the litter decomposition of rate of litter decomposition in forest floor of Similpal Biosphere Reserve, and why that only indicates the microbial population. The microbial population is more in the undisturbed sites and then in the moderately disturbed sites, and very least in case of disturbed sites. Because I am not presenting here the litter decomposition data or the microbial population data of Similpal Biosphere Reserve. But in case of available phosphorus, what happens in case of available phosphorus? In case of available phosphorus, there is not any type of trend is marked in case of available phosphorus. And when we can go to another chemical parameters of the soil, that is known as the available potassium. The available potassium content, of the soil of Similpal is decreasing from the disturbed one to the undisturbed one. And here the question arises, why the available potassium content is decreasing from the disturbed sites to the undisturbed sites? The most remarkable reason is that is the forest fire. So during forest fire, what happens? The litters are completely burned out, as well as also the different lower groups of vegetations, that is the some of the shrubs, some of the herbs are also completely burned away. As a result of which, what happens? There is the deposition of ash. When there is the deposition of ash, that ash is leached, and that ash is also deposited over the soil surface on the forest floor of Similpal. As a result of which, due to ash deposition, the potassium content is more in case of the disturbed sites, because forest fire is more in case of disturbed site. But here the question arises, somebody wants to know that whether that forest fire is natural or the forest fire is unnatural or not natural. The forest fire is not natural one here, because that forest fire is man-made. And why the forest fire is there? The forest fire is only to pluck the flowers of mohua, that is known as the Maduka indica. To collect the mohua flower, the local population, they provide fire on the floor, forest floor, and when all the vegetations along with the litter is completely burnt away, then the surface is clear and they can collect easily the mohua flower. As a result of which, maximum amount of, due to forest fire, maximum amount of ash is deposited over the soil surface, that leads to the higher value of available potassium in the disturbed sites in comparison to the undisturbed and moderately disturbed sites. Now we can come to another chemical parameters of the soil, and what is that? That is the C/N ratio. The C/N ratio, the carbon to nitrogen ratio of the soil, indicates about the soil fertility status. So if the C/N ratio is very high, then soil is not fertile, and if the value is low, then we can depict that the soil is a fertile soil. So here from the data, what the data indicates? The data indicates that in case of the CN ratio is decreasing from the disturbed site to the undisturbed site, that indicates the soil fertility across the disturbance gradient is increasing, as well as also along with the depth, the fertility status is decreasing. Why the fertility status is decreasing and the CN ratio is increasing with respect to the depth? Because if the litter is decomposed over the soil surface, then that the humus is deposited and that is on the soil surface, and the soil surface having more organic carbon, more nitrogen, and more total nitrogen, as a result of which the surface soil is more fertile in comparison to the subsurface soil. So after the soil, now we can come to the biological attributes or the biological parameters of simple biosphere. And what are the different biological parameters? The biological parameters or the different phytosociological attributes are indicating about the condition of the forest cover. And here the biological parameters are the density, the basal area, the species diversity, that is known as the Shannon-Weiner diversity index. Another one is known as the concentration of dominance. So here the density of the two different layers, that is known as the three different layers, that is a tree, shrubs and herbs, or the seedlings, shrubs and saplings. And here we can measure the tree density, shrubs and sapling density in terms of plants per hectare, and the density of the herbs and seedlings in terms of individuals per meter square. So here the plants are divided into two different groups, that is known as the dominant species and all species. So in case of tree layer, for the dominant species, the density value increased from the disturbed to the undisturbed sites. For the all species, the density value is also increased from the disturbed site to the undisturbed sites. And in case of India, in case of study of the density in different parts of the Indian forest covers, and what is the variation in the value of density in different parts of India, that value varies from 298 to 986 number of plants per hectare, as per the study carried out by Kitamura et al. 2021. In very recent study that indicates the density value varies from 298 to the 986 number of plants per hectare. So our study also indicates within that range. So that is a maximum value is 920 for the all species, and the minimum value is 655. And for the dominant species, that is 435. And here the value for the dominant species 355. But now the question arises, in case of a dominant species, why the density is less in case of undisturbed sites? And because which are the dominant species in case of simple biosphere reserve, like the Soria robusta, Terminalia alata, Terminalia cebula, Terminalia belirica, that is Phyllanthus emblica. Another one is known as the Anogenesis latifolia. All are known as all the tree species are the light demanding species. When maximum light is available on the forest floor, then their seedlings are grown up, easily grown up to the sapling stage, then to the bolt stage, then to the post bolt stage, and then into the higher category of girth classes. As a result of which, low light is available in the undisturbed sites, and in comparison to disturbed sites, so the density of the dominant plant species are less in case of undisturbed sites and high in case of disturbed sites due to high light intensity on the forest floor. But in case of all species data, what it indicates, the density of plants per hectare is more in the undisturbed sites and less in the disturbed sites. But in case of shrubs and saplings, and in case of herbs and seedlings, the reverse trend is marked. Why the reverse trend is marked? Because only depending upon the light intensity on the forest floor. In case of a basal area, same trend is also marked in case of basal area. In case of basal area, for the all species data, this is known as all species data, but this is known as dominant species data. But here, in the dominant species, one of the plant is marked that is known as the Soria robusta. Soria robusta is one of the economic plant, and that is used for various purposes. If it is used for various purposes, in case of the disturbed sites, the people are lopping the Soria robusta plant, as a result of which the value of a basal area of the dominant species are less in the disturbed sites and more in the undisturbed sites. And for the all other species, that is more in the disturbed sites and less in the undisturbed sites. And similarly, in case of species diversity, the Shannon-Wiener species diversity value, that is more in the undisturbed sites and less in the disturbed sites, because more number of species are available in the undisturbed one and less number of species are available in the disturbed one. And when we can concerned about the another diversity index that is calculated by Simpson, that is known as the concentration of dominance. So these two parameters are very closely related to each other, and these two parameters are inversely correlated to each other. If species diversity is more, concentration dominance is less. If concentration of dominance is less and species diversity is more, because the site is more diverse and the site is less dominance. The site is less diverse and more dominance. So as a result of which, such type of phytosociological attributes we have calculated for the different plant life forms or the different vegetation layers of similpal, that is tree, shrubs, and saplings, herbs and seedlings in different disturbance gradients of similpal. That indicates the health and the wealth of the forest cover of similpal biosphere. Next one. So gradually, day by day, what happens? The population level is increasing, or if the population level is increasing, the forest cover is not increasing. Rather, the natural forest cover is decreasing. Why? I want to tell you one thing. That is, we can worship the invisible God, but we can destroy the visible God. And which one is known as the visible God? That is, nature is known as the visible God. And we can destroy the different varieties of the plants for our own benefit, for our economic benefit, and we also destroy the different types of the economic plants to make different types of furnitures, or by means of the timber poachers, they can taking away the valuable economic plants from the forest covers and sold it in the market and getting maximum economy. So in this context, I want to tell you and how we can know about the condition of the forest cover and what is the state of regeneration of the tree species in different study sites of similpal biosphere. Here there are five different types of regeneration categories are there. And what are those five different regeneration categories? And that is known as the good, fair, poor, none, new. And why the regeneration is called as good, regeneration is called as fair, regeneration is poor, regeneration is none? Because that is depending upon the three different stages of the plant life. And what are the three different stages of the plant life? That is, one stage is known as the seedling stage, next stage is known as the sapling stage, and next stage is mature one. And by taking the 3 different stages of the plants, we can calculate the regeneration potential of the tree species in different study sites of simil palm. And first one is good, and when we can told it as the good condition or that is good in regeneration, because if the seedlings, number of seedlings are greater than sapling and greater than adult. Now the question arises, which one is called as the seedling, which one is called as the sapling, and which one is called as the adult one or the mature one. So that classification is depending upon the GBH, that is known as girth at breast height. And another question arises, when we can measure at what height we can measure the girth of the plant. We can measure the girth of the plant at 1.37 meter from the ground, that is 137 centimeter from the ground. And when the GBH is less than 10 centimeter, then that is called as the seedling. And when the GBH is 10 to 30 centimeter, then that is called as sapling. And when greater than 30 centimeter, that is considered as the adult one. So the condition is said to be good if seedling number of individuals in the seedling category is greater than sapling category and greater than adult, then that is condition is called as the good regeneration. And when it is called as fair, when seedling population is greater than sapling population and the sapling population is less than equal to adult population, that is called as the fair regeneration. And poor regeneration, the state is said to be the poor regeneration if a species survive in only sapling stage but not as seedling and not in case of the mature stage, then that is called as the poor regeneration. Because no one is there, if seedling population is not there, then how it can transfer to the next higher girth class. And the fourth one is known as none. When the regeneration is said to be none, if a species is absent both in sapling and seedling stage but present only at the adult stage, then that is called as the non-regeneration. And when it is called as the new regeneration, if a species has no adult but only saplings and or seedlings are there, then that is called as the new one. Because if the adults are not there, only seedlings and saplings are there, then that may be migrated from or due to seed dispersal or due to any other way, the plant is newly regenerated. That's why that is known as new regeneration. So when we, hello? When we
Continue.
Hello?
Continue, sir, continue.
When we go for the study of regeneration status of different types of regeneration status of plant life forms in different sites of simil-pal biosphere reserve, it indicates that the total number of species in 5 different categories are high in case of the 5 different categories and their percentage. Then we came to the one type of or we can take the management strategy, or we can draw the conclusion from here. And what type of conclusion, depending upon that table, we can summarize it. That is the regeneration of species in the 5 different growth forms in different groups of the plants, that is large trees, medium trees, small trees, shrubs, climbers. And that is the total numbers of species, and this is the percentage. And when we can find out the percentage, the plants having good regeneration is 22%, and fair regeneration is 11%. That is both ones are favorable ones for the forest covers that summed up together to form 34%, and the poor, none and new, that is more than 50% or more than 60%. So it indicates that the forest condition is in the forest condition is in the poor state of management. From the regeneration status study, it indicates that the forest of the Similpal Biosphere Reserve is in the poor management strategy with respect to regeneration. Now we can come to another parameter, phytosociological parameter, which is of great importance. That is known as the importance value index. And why we can take the importance value index? Because we can measure the density, we can measure the vessel area, we can measure the frequency. The frequency indicates about the distribution. The density about what is the density of the plants per meter square or per hectare, how many number of individuals are there. Basal area indicates how much coverage it can cover. But the three different phytosociological parameters, that is the density, the frequency, and the basal area, they can, such parameters can indicate the different types of conclusion. But when we can mix it together, when we can sum up all the three parameters, that is the density, the frequency, and the basal area, in terms of relative frequency, relative density, and relative basal area, then we can find out, or we can calculate one of the index that is known as importance value index, which is more appropriate in comparison to the calculation of the density, frequency, and basal area. And these 9 dominant species of the Similpal Biosphere Reserve in 3 different disturbance gradients. What is their IVI value or the importance value index? Albizia lebec having the lowest one for all the 3 different disturbance gradients. Then Carya arborea, then that is the Diospyros embryopteris, and the maximum importance value index was sought by the Soria robusta, then all others, Terminalia alata, Terminalia valerica, Cassia fistula, Phyllanthus umbilica, and Dalbergia sisu. They have very less importance value index in comparison to Soria robusta. Because Similpal Biosphere Reserve is one of the biosphere reserve where the sal plant is the dominant plant. And why sal plant is the dominant plant? Because the soil condition and the environmental condition is favorable for the growth and development of Soria robusta. Not only the soil condition and environmental condition is favorable for the growth and development of Soria robusta, because the Soria robusta is such a plant which can regenerate through their roots. That is known as a root sucker. Because there are 2 different types of populations are there. One is known as the ramette population, another one is known as the genet population. So what is genet population and what is ramette population? Genet population, when the plant is germinated from the seed, that is called as the genet population. And what is ramette population? Ramette population, when another plant or new plant is developed from the root of the original plant, then that is called as the ramette population. And mostly Soria robusta in Similpal are showing ramette populations are more in comparison to the genet population. And though the ramette population is more and the Soria robusta having higher IVI values, higher density, higher frequency, and higher basal area in comparison to the other 8 different dominant plant species of Similpal. Now we can come to the ecological aspect of the ecology and uses of the 9 dominant tree species of the Similpal Biosphere. Why we can consider about the 9 dominant tree species? The 9 dominant tree species that is based upon the IVI value. We can select these 9 species because their IVI value is nearly about 50% of all other plant species. So due to this reason, we have to select these 9 tree species as the dominant tree species. And what is their ecology? So some are deciduous, some are moderately tree, some are light demander, drought resistance, fire resistance, canopy tolerance, fire tolerance, and salt and shade tolerance. And these plants are used for different economic purposes as well as also the wildlife purposes and pharmaceutical uses. Though the plants are utilized for economic purpose, wildlife purpose, as well as also pharmaceutical purpose, then so all the 9 species, what is their economic use value? We have to calculate their economic use value. So all the different species having different types of use value. So this is the use value, and how we can calculate the use value of different plants. The use value index is equal to UI divided by N. And what is UI? The number of uses mentioned by each informant for a given species. Suppose for Soria robusta, suppose we can take number of informants is 10. So each informant telling about Soria robusta is utilized for which purpose? First informant tells that Soria robusta is utilized for 3 purposes. Second one is for 4. Third one is for 2. Then we can sum up all the number of uses mentioned by each informant for a given species divided by the total number of informants. Then we can calculate the use value. So from this use value, it indicates that the Soria robusta is having maximum use value, followed by Terminalia valerica, Phyllanthus emblica, Dalbergia sisu, Cassia fistula, Terminalia alata, Albizia lebec, Carria arborea, and Diospyros embryopteris. And this Diospyros embryopteris is one of the evergreen species, and all others are the light demanding ones. So based upon this, so what it indicates, the Soria robusta is mostly utilized by the people who are remain inside the forest or who are also present outside the forest. So our prime aim to provide conservation strategies for such type of plants. So this is how the different types of plant species are utilized by the forest dwellers. That is Eulaliopsis vinata, that is sabai grass is used, that is the flower of Maduca indica, that is the root of Asparagus racemosus, that is Phyllanthus amarus, and the bark of Saraca suca, that is known as Gymnema silvestri, and Piper lungum, and the rhizome of different medicinal plants that is collected by the Khadia people, and they can sold it in the market and get their livelihood or economy, and the collection of different types of NTFs, non-timber forest products, by the tribal population of Similpal from the Similpal Biosphere Reserve and sold it in the nearby market, and they can earn their livelihood. So that's why we can calculate the use value. So now I am come to the another part of my study, that is known as the population structure of the different varieties of the plant life forms of Similpal Biosphere Reserve. And why population structure? The population structure can predict about the future of the forest covers, whether in the future the forest cover will remain or in the future the forest cover will disappear. So that can be modeled out through the study of the population structure. So in case of population structure, that is taking all species into account and the dominant species into account. So when we can study about the population structure, different age groups are there. That is the juvenile, young, elder, mature. So like the human population, like the different stages of human life, starting from the birth to the death, like the Shakespeare's drama, the different stages of life. Similarly, in case of plants, there are different stages. What are the different stages? That is the seedling, sapling. The sapling is known as the juvenile. Next one is known as the young. Next one is elder. Another one is mature. And the juvenile, young, elder, and mature, that is depending upon the GPH. So when 30 to 60, that is called as juvenile. 60 to 90, that is young. 90 to 120, that is elder. Then greater than 120, that is called as mature. So in case of, by taking the number of species in each group or in each age class, that is the number of species are decreasing. Similarly, if we can take the density, then that is also decreasing from the juvenile to the mature one. That performs a L-shaped graph. That indicates the good condition of the forest cover. In the future, the forest cover is retained and survived. But in case of a basal area, the number is less in case of a mature, but basal area is more due to high earth class. That is also a good indication or sign for the similpal biosphere reserve. Similarly, in case of dominant tree species. Next, we can come to the population structures in three different disturbance gradients. In case of disturbed sites, what we have marked, that is the density is maximum in case of seedlings, but zero in case of mature one and elder one. Why? Because due to looping practice, the people are cutting away, or people are cutting up the higher girth class plants for their use, as a result of which the higher girth class individuals are not appeared in case of disturbed ones, because this bar is responsible for density and this is for the basal area. No basal area. Basal area is very, very minimum. So in case of moderately disturbed sites, perhaps that is very similar to that of the disturbed ones. But in case of undisturbed ones, the basal area is more in comparison to the disturbed and undisturbed one. Why? Because the higher girth class individuals are not cut away or not lobed by the individuals, because the undisturbed sites are mostly located in the core zone of the biosphere reserve, which is declared as the tiger reserve area, that is 2,750 square kilometer area. And to the tiger reserve area, no human habitation, nothing else, no such type of disturbances are there. So from these three different diagrammatic features or three different graphical representations, what we can indicate or what we can conclude, in the future, the forest cover is survived if only can provide the protection, proper protection to the forest cover. Then another parameter was also analyzed for the 9 different tree species, that is the relative growth rate of the plants. Because nowadays what happens, what the forest officials can do, suppose there is some type of a degraded areas are there, they can plant eucalyptus or they can plant eucalyptus. And when they can plant eucalyptus, and another one is known as the Alstonia scolaris, and why they can put such type of plants in the degraded areas, because such plants having high growth potential, because and having no economic use. So as a result of which, if we can study about the germination potential and the growth rate of the indigenous ones or the dominant plant species of this similpal biosphere reserve, and in which conditions they are survived well, If we can put or planted such indigenous varieties, then that can attract the different types of wildlife, and there is the plant animal interaction, and the forest cover is well maintained, and the biodiversity potential is also well maintained. So when we can calculate the absolute growth rate of the 9 different dominant plant species of the reserve, all out of the 9 different plant species, the Alpigia leve, Carya arborea, Phyllanthus symbolica, Soria robusta, Cassia fistula, Dalbergia sisu, Terminalia alata, Terminalia bellerica, all are having higher growth rate in the disturbed sites in comparison to undisturbed sites. Why? Because all are the light demanding species. If in the disturbed area any type of degraded conditions are there, if we can put such plants or planted such plants, then there is the better growth of such type of plant species, and that can attract the wildlife, as well as also increase the biodiversity potential of the similpal biosphere. But in case of Diospyros embryopteris, which is a semi-evergreen one, and which is a semi-evergreen one that can having maximum growth rate in the undisturbed sites in comparison to the disturbed and moderately disturbed sites, because such a plant is well developed in the shade conditions, and this is known as a shade tolerant plant, and that the products of the Diospyros embryopteris, the fruit is mostly utilized by as a food material for the monkeys. The fruit of Carya arborea is utilized by the elephants as the best food. The bark of Terminalia alata, Terminalia bellerica are also utilized. Cassia fistula is used by different types of animals, and all the 9 different species are good for different types of frugivorous birds, as well as also to attract the different types of animals of similpal biosphere region. So how we can regenerate such type of plant species in the field conditions? So this is the germinating seeds of Diospyros embryopteris, Carya arborea, that is seeds of Terminalia bellerica, that is the seed of Terminalia alata, seed of Soria robusta. Because here one of the peculiar character is there. When the seeds of Soria robusta is fallen on the forest floor, and that is immediately attacked by the fungus, so the germination potential is reduced. So we are developing one of the method how to germinate the seeds of Soria robusta within 3 to 4 days, and this is also the germination activity of or germinating seeds of Phyllanthus symbolica. And gradually how the plants are grown up, the seedlings are developed, seedlings of Diospyros embryopteris, seedlings of Cassia fistula, seedlings of Phyllanthus symbolica, seedlings of Carya arborea, Terminalia alata, and Terminalia bellerica, what we have developed in our garden, university garden. And ultimately, what is the conclusion? What is the conclusion of my study? And my study mostly divided into 2 different aspects or 2 different stages. One is the monitoring part, another one is known as the assessment. In the monitoring part, we can inventory the forest ecosystems, identified the biodiversity rich pockets, and site quality assessment by the calculation of disturbance index and Ramkrishnan index of stand quality, and how we can go for assessment. In one side, we can measure the relative growth rate of the plant species, whether that species is having adequate growth, then that is suitable. If that is not adequate growth, then again we can come to the measurement of the relative growth rate. And in the next part, we can go for the demographic analysis, that is the population study and the phytosociological analysis of the plant species and the regeneration study of the plants to their higher girth class or the higher age class. If that is okay, then if that is possible and that is in a good condition, the regeneration and the population structure is in the favorable condition for the forest cover, then we do not then we know that the forest cover can regenerate easily and can survive in the future. But if no, then further we can go to the same type of study. And I have selected 9 species and documented out and carried out such type of assessments of the 9 different species, and out of the 9 different species, 8 are favorable for the disturbed and moderately disturbed site, and 1 is for the undisturbed site. If such type of species are planted in the degraded conditions of the Similpal biosphere reserve, the biodiversity potential of the Similpal is well preserved for the future, and the people who are living inside the forest, as well as also the people who are survived outside the forest area, they are benefited, and the complexity or the complex ecological condition of Similpal biosphere reserve is retained and maintained for the future and for the future generation. So such type of studies are highly essential. Thank you all.
Thank you, sir. The forum is now open for doubt and discussion. Any questions? Okay. So our next resource person for today is Professor Vidyanath Jha, Principal and Professor of Botany, LN Mithila University, Darbhanga, Bihar. On to you, sir.
Yes. Yes. Go ahead. Still sending.
Dr. Mishra, sir, you'll have to stop presenting, sir.
Okay, I am stop presenting.
Okay, sir.
So, welcome everybody. Am I audible?
Okay, sir.
Yes, sir.
I am audible, yes. Good. So the topic of my presentation is utilization of components of biodiversity in the floodplains of North Bihar. Now this has been organized by a number of collaborators in this conference on biodiversity building and natural resource management. Yes. From the extensive lecture by Professor R.K. Mishra on forests, I would like to bring you to the world of aquatics. I am talking about North Bihar, that has a large number of stagnant and flowing water channels that constitute a repository of aquatic biodiversity. Water plants like Makhana, that is Urel ferox, Singhara, that is Rapabaia spinosa, Khubhi, that is Cirpus articulatus. Its name has now been changed to Chenoplectiella. Then there is Bhent, that is a Nymphaea species, and a host of other plants provide a basis to the livelihood of people in this area. All efforts are being made to make Bihar self-sufficient in fish production. Of late, fish farmers are practicing Makhana-based integrated aquaculture to facilitate the success of blue revolution in the state. Species of Nelambo, Astera cantha, Ipomia, Alternanthera, taifa, etc., are also utilized by local people
Sir,
for the various requirements of livelihood.
sir, are you presenting slides?
Yes, slides are there. This is the
No,
first
no,
slide.
no presentation.
No
Just
presentation.
share slide.
Share karo. Slide share karo. इसके बाद आओ तो
Now visible.
next. Now visible. So
Slide, sir. First
local
slide.
people. I have
So,
talked
first
about. I have talked about a number of significant plants, species of Nelambo, Astracantha, Ipomia, Alternanthera, typha, local people for the various requirements of livelihood. One is Skinomene, belonging to the traditional gardening community for Kodhila art. Then there is Sesmania species. They are proving instrumental in the success of second green revolution in Bihar. Local farmers are also using water hyacinth, that is Icornia crassipes, under the Jhang method of capture fishing. Vetiver grass is being cultivated for aromatic oil as well as for land conservation. It provides a basis to the famous Sikki art of Mithila region. The plant has attracted a wider scientific attention in view of its potentials for tackling the menace of global warming. Flood plains in North Bihar also harbor the luxuriant growth of palm species like Phoenix dactylifera, Borasus flavellifer. In certain pockets, Bisar, that is seedlings of Borasus flavellifer, cultivation has taken the shape of a cottage industry. So with this, I would like to go to further. Yes, next slide. Next slide. Now, North Bihar has a fine network of lotic and lentic water bodies. Now, on the left side, there is a picture of the various rivers that form a network, fine network of rivers and rivulets. And on the right side, there are 27 ponds in a ring in a village of Madhubani district. That is, this is land record, and you can see how the 27 ponds, they are arranged in a ring. And there is one river called Sugarbe River that feeds all these ponds one by one. The practice, centuries-old practice, is still there. It's still serving its purpose. And then there are the ponds showing the important bird sites, and then other water bodies. Next one. Now, I have to talk about the aquatic phytodiversity first. So there are a number of plants, as I have already told, Tal Makhana, then Kodhila, Sarhanchi, Motha, Jalkumbhi. These are the local names, and the plant names are also there. On the left side, Astracantha, then Skinomene, Alternanthera, two species, Phylloxeroides and Sessilis, Cyprus rotundus, then Ipomia aquatica, Ipomia carnea, Nelambo nucifera, Nymphea alba, Nymphea nauchelai, Nymphea stellata, Cirpus, other species of Sesbania, Trapa, Taifa, Vetiveria. The name of Vetiveria has now been changed to Chrysopogon digenoides. Now these are used for various livelihood purposes. Astracantha used as leafy green source of, then Skinomene is used as biofertilizer, then Kodhila art, then Alternanthera phylloxeroides used as leafy green, then also as fodder, Alternanthera sessilis, again leafy green, fodder, and also as fish shelter belt. Then Cyprus rotundus, its scomes are used for making shital party, that is mat. All these are now yielding to the production of the synthetic ones, but even then, Icornia crassipes, it is Jalkumbhi, found in wild form, used for serving the purpose of energy, then fishing shelter belt, then fodder. Then I would like to talk about Udal ferox, on which I have worked, and it is in cultivated form in our area. It is used as subsidiary food, and component of integrated aquaculture. POPs are a ritual item. Now also a significant immuno, that is karmi sag, used as leafy green. Then Ipomoea carnea, that is called as patit or behaya, as it grows anywhere and everywhere in the water body. It's used as fuel wood. Nelumbo nucifera, it is also used as subsidiary food. Flower and seeds are ritual items. Nymphea alba, subsidiary food. Nymphea nauchela is subsidiary food. Flower used as a ritual item, source of drug. Then Nymphea stellata, subsidiary food. Flower a ritual item, again source of drug. Cirpus articulatus as subsidiary food, so on and so forth. There is a long list. Now this typha, it is used as pig fodder. Leaves used for mats. Silky florets used for stuffing. Khadi Gramadyog used its florets, silky florets for stuffing material. And then Vetiveria gigantoides used as flood fighter, fence item, then also in Sikhi handicraft. Next one. Now this slide, which includes a number of makhana items, shows on the left, right, left above, these are the makhana pops. And then I am showing you the picture of Professor Nama Goren Inbar of the Institute of Archaeology, Hebrew University of Jerusalem, Israel, who made a visit to Darbhanga in September 2011 to know the details of the cultivation and utilization processes of gorgon nut. Her visit was in light of the fossils of 2 aquatic nuts, including makhana, it's called as gorgon nut or fox nut, and singhara, that is water chestnut, along with 5 other terrestrial nuts having been discovered from the Herculean site of Jesher Benot Yaakov in Israel, along dating back to the early Middle Pleistocene. So this was with this purpose that she visited Darbhanga. And then we have in the middle a photograph, a picture showing its distribution in India. You see, its wild populations are available from the north western Kashmir, temperate lakes of Kashmir, and then its wild population in the entire Himalayan belt, and then also in Assam and Manipur, other northern district Tripura. These are the states where its wild populations are found. Now its cultivation is localized in 8 to 10 districts of our North Bihar, that is Mithila area, and at all other places it is available in wild form. And people, basically the Adivasis in around the Loktak Jheel of Manipur, they collect it from the, and it is sold. Fruits are sold as such in the Ima market of Manipur. So they want that like it is done in North Bihar, Mithila area, there should be a harvesting process from the pond, because they are simply plucking it by ferrying it on the boat. And the practice that is there and practice that is here, both these should be amalgamated, and that would provide a boost to this makhana. On the left side, this one, and this shows how makhana is deeply enshrined purposes. Here you can see this is for medical purpose, and the betel is also there, and makhana is also there, other items are also there. And so on the here we are showing that 8 to 10 districts in North Bihar where it is being cultivated in the ponds, the very deeply intricate process of its cultivation. And in the middle down we are showing a picture, a figure of the big pond of Karnal Dynasty pond, which shows how integrated aquaculture is being practiced. Open space is for the this air breathing fishes, water breathing fishes, and for their sustenance. And it is on account of this, there is nothing except seed is taken out of the system. So the entire area which is covered by the leaves, dense udal leaves there, and the detritus is there on the bottom, and detritivorous fishes like singhi, mangur, kabai, etc. They survive on the detritus, so they are this detritivorous fishes, and we have water-breathing fishes that survive in the open space. So this monotypic genus Udal ferox has a discontinuous distribution in freshwater pools of northern India, northwest temperate lakes of Kashmir, and northeast Himalayas, and states like Bihar, Assam, Odisha, West Bengal, including Bangladesh, Tripura, pockets where wild and cultivated populations of Udal ferox are. Now, next to Makhana, I have Singhara, that has not been shown here. Singhara, that is Trapa natans variety by Spinosa. Earlier, so far as the biodiversity loss is concerned, and the Quadrius spinosa was available during our childhood days. Now that Quadrius spinosa gave way to the Spinosa, and now there are spineless varieties of this Trapa natans. They are now available, and I think next to Singhara in our area, it is the Cirpus articulatus, whose name has been changed to Chenoplectiella articulata. Now this is the basis for the basis of Khubhi Ramdana industry, because it pops, very small pops. They resemble small pops. They resemble those of the Ramdana, that is amaranth, which is a land plant. So in our area, it lavishly grows in the floodplains, which are waterlogged. So this shows us the story of how these granular pops are made, and they are sold very lavishly. And on the right upper side, we have its seed coats. These coats are used for making the earthen houses to give strength to the mud that is used for housing purposes. And here, these are the infructescences. These are called infructescences, in which from which the seeds are taken out, and these seeds are just this is the granular sweet made out of the pops of this Cirpus articulatus. So next one. Yes, I will come to it later also. This gives us an idea about Makhana-based integrated aquaculture. Just to see this round figure shows how the stages of fish and Makhana. Yes, this is sowing stage, then germination, then membranous leaves are there, then there is flowering and fruiting, and then bursting of fruits, then harvesting. So year-round cycle that accommodates the settling of carp and air-breathing fishes that is there. And on the right hand side again, another pond, another big pond in which open space has been left out in the middle to facilitate the integrated aquaculture, that is Makhana-based integrated culture, to make this blue revolution a success. All the efforts are being made in our country. Next one. So there are a number of fishes available, like this rohu, then other pothia, nani, etc. The big list, reva, etc., etc. So these are the various fishes, and their diversity, their biological names are also there, and their range is also pricing range is also there. So this area was earlier known for its fish diversity, just like paddy diversity. Fish diversity also was very much extensive in this area, but now with the depletion in the water body, depletion in the amount of water body, and in the amount of water body, there has been a great loss to the fish diversity. Next one. Now, fishes reported from Makhana ponds, I have already told: Channa punctatus, Heteropnistus fossilis, Clarius batrachus, Puntius sophor, Anabas testudineus, Channa striatus. So these are the fish species which are abundantly available in the Makhana ponds. Next. Now, next to this Makhana, another plant that is of much use so far as the land conservation practice and amelioration of the soil quality, that is vetiver. Just see here how a woman is taking out its peduncle that forms the basis of the Bihar tableau, how this Sikhi art is there, prevalent in our area. Down and on the left side, these are the inflorescences that are collected at the end of rainy season by the local people who derive their livelihood by carving beautiful containers, Sikhi containers, by the rural poor people. Next one. Now here see, this vetiver, that is Azotis vetiveria gingivitis, a multifaceted wetland grass with a role in meeting the challenge of climate. How alongside the railway line, it's there. Then a number of other products here. See, and this is very, I want to stress upon, is that in our area there is a practice in which this vetiver grass is formally worshipped by women in the rural areas where this animal rearing is still prevalent, and it is worshipped along with the cow dung and the soil cast. So this provides look towards other countries for citing examples of land conservation practices. Here we have an indigenous practice in our area that shows us how this multifaceted grass used extensively in our agriculture or in horticulture practices that is being worshipped. So this is a fine example as to how our soil health can be maintained by maintaining this vetiver cultivation. Now here on the right, on the middle, beautiful brooms are being carved out of this katara jhar, that is it's called. Then we have chappals, then we have hats, and just to see on the right upper side how alongside the river it grows. It grows in a self-dependent way, and it brings out a halt to the landslide, soil erosion at the time of the floods, high floods in the rivers. And down right side we have just to see Sikhi Taj Mahal made by a local artist adorning the Darbhanga Museum. Now the beauty of this art figure is that it completely evades the attack of pests like fungi, insects, etc., etc. Now this beautiful item is still there in the Darbhanga Museum, right from the time of Darbhanga Raj. Next one. Now just see, cultivation of vetiver in Madhubani wetlands. Now this is a progressive farmer, Lal Bahadur Singh. See how he has just made an Indian of this eucalyptus along with this vetiver. Now this timber is relatively grows as compared with other timbers. It grows lavishly and is able to provide logs to the needy persons who cannot afford the timber items. So this fills the gap in the availability of. Here on the upper side, this is in a mature state. Now people are cultivating it for their aromatic roots, and the beauty is that it withstands the and the sands alongside the rivers on the other side. So this provides us a very good example of how vetiver can take us out from the morass on account of high level of water logging in our area. Next one. Now just see again innovative vetiver wall hangings. They are carved out of. We have two types of weaving practices, making items from this vetiver. One is through weaving, and another is through cutting and pasting. So here, an innovative vetiver person, he has been able to just carve through cut, bend, and pasting method. This is a new innovation, and Sri Dhirendra Kumar in Rampur village of Pandaul block of Madhubani, he has made it, and the government has by raising common facility centers based on this type of vetiver innovation. So this the phytodiversity in wetlands could be put to a diversified use through art practices also. Next one. Now here we have the example of eskenomine, which is in our area it is called as Kodhila, and it is a potential biofertilizer in deep water paddy fields of North Bihar, and is a basis of Kodhila handcraft. Now here we have profuse stem nodulations over its stem, as this is a natural adaptation in this plant. The nodules are there on the stem that remains above the part that remains above the waterlogged situation, and paddy field. And these are the spots, round spots, where this eskenomine grows naturally along with the sea. Yes, this one, and again this one. This is how the logs are there, and then current science, our journal, leading science journal, had a picture of it. That is this picture of this goddess that is built of this Kodhila, log of eskenomine species. And here again, see just how it is offered to the tree gods. So this is an example of how eskenomine, which is a fabaceous plant, can lead to the it leads to the function of nitrogen fixation, and it could be harnessed on a larger scale for raising the fertility of the soil. Next one. Livelihood. People belonging to the Mali caste in our area, they carve a number of items. In Bengal, it is called as sola wood, one of the lightest wood known. Various items are carved out of it for offering in the temples, etc., etc. Next one. Just see. Now we are coming to the nuisance weed, that is Eichhornia crassipes. Now it came to India about a century back, and earlier it was not here, but in the tropical waters, water bodies of this our country, it infested almost all the water bodies. And just see what the situation is there. On the left upper side, these are to herons, and these are the jhangs that are raised for capture fishery in the water bodies. So picture providing picturesque this scene, how the jhangs are being raised. Next one. This pankawa is there, and then people in Kusheshwarasthan area, they are burning it, and the ash is being used as fertilizer in the maize fields through the digging method. And here they are collecting the fishes from the jhangs. And here on the right side, see how this Eichhornia crassipes, leaves of Eichhornia crassipes, they are being harnessed, they are being chopped off for being used as this fodder by the animals. And on the middle side, you can see how the animals, they feed upon, and this they are free, they are roaming in the water body and are feeding upon this water hyacinth, and the mollusk eggs, they are there on it, and that way that transmits the diseases like this mollusk-borne diseases, and they fall prey to it, and this way they just suffer from the diseases, mollusk-borne diseases that are there in the water hyacinth. So Eichhornia, it is a major hindrance to agriculture and culture, and an innovative model of hyacinth-based capture fishing called jhang, developed by local people in chores, stagnant water, witnessing extensive growth of submerged weeds. A method also applicable in the rivers or rivulets after the current speed becomes slow. Normally it is in stagnant water, but even in the rivers, these jhangs are raised. System provides a sense of respectability to the otherwise difficult to tame hyacinth. If we cannot tame the hyacinth, then we should go for harnessing whatever is good in it, which is one of the 10 most obnoxious weeds of the world. hyacinth ash applied to manure as manure to the crops, and mixed soil beds of summer paddy. That practice has now diminished, but it was there. Beds were prepared in the soil. Now here just see another innovative practice related with hyacinth. Hyacinth being used as bridge over a river during flood. Just see this floating bridge made of this water hyacinth, Icornia crassipes, in the Kamla River in Birol subdivision of Darbhanga district. This was there in August, September 2011. So this is an innovative practice. People have no way out. Even today, after so much of progress, so much of development, people have to resort to these practices, whatever is available with them at the moment. During extreme floods, when there is no way out, they have to resort to a practice like this. Next one. Just see. Now we are coming to Sesbania species. Now we have a number of Sesbania species, and Sesbania rostrata, which is called as manager in Kusheshwarasthan area, wetlands, which are protected areas, emerging as lifeline in the floodplains of Kusheshwar. How young girls, they are collecting its fronds for being fed to the goats and other animals. They have no way out. They are just ferrying over the rivers. Young boys and young girls, they are doing it during extreme floods. There is no way out. Now this manager fronts, they are being fed to the animals as fodder. Next one. Just see. This is how a practice, innovative practice that is there. Farmers in eastern city blocks of the district in Birol subdivision are growing manager, that is Sesbania rostrata, as a multifaceted plant which serves the purpose of fodder, fuel, and low-cost housing material. It has proved a flood fighter as well. Now its seeds are also being fed to the cattle and are used as a substitute. This also provides an example of how a plant is domesticated. Another species of Sesbania called dhaicha is being spearheaded to be grown as a biofertilizer, for the sale of dhaicha seeds to the farmers. Now Bihar is fast emerging as a citadel of the second green revolution, and the government has taken on a large scale up the distribution of this dhaicha seeds to the farmers. There was a practice earlier between Rabi and Kharif crops that this plant was grown, and it was simply just made to fall flat on the ground. Now here this tractor is being used for that purpose. Earlier it was done through auction, and the practice has again been brought to life by the government, which envisages to bring Bihar a platform, a citadel of the green revolution. Next one. Moong is also distributed. Now the next plant is called in our area as thethar or behaya, meaning thereby that which grows on its own, a biomass plant with a role in checking soil erosion also. Now just see, this Ipomia carnea is there alongside the bank of a river. Now it checks the spread of soil erosion. Now here it's biomass, dried thethar used as fuel. Now it is obnoxious, its smell is obnoxious, but poor people, they have no option, they are doing it. Now here again on the low behaya planted as a fence alongside a new earthen road for its protection from erosion by high floods. Now these earthen roads, they have to develop, a mechanism has to be developed for protection against high floods. Now to an extent, this Ipomia carnea provides protection against the current gushing current of the flood water. So here this is an example of how Ipomia carnea is being used as a fence purpose. Next one. Now here, capture fishing, still a way of life. Now these are the various fish species, air-breathing fish species. This is mangur, that is called the state fish of Bihar. Then people are doing angling. Angle fishing is also a way of life in Bihar. It has been made free. Government has made it free. People thrive upon this angle fishing. They are doing it. Just see. And we have also the crabs on sale during peak flood years. This is a thriving business. The crabs are there. then the mollusks are all these aquatic biodiversity, animal biodiversity that is being used for livelihood purposes. So next one. Now here again, we have an example of at least 100-year-old, a century-old pearl button industry in the Mehsi of East Champaran district, Mehsi town or sub-town of East Champaran district. Observe, see, in the Sigrahana river, a precursor of the present Buri Gandak river, Paresia favidens. It is a source of pearl button industry in Mehsi. Now just see a number of ornaments, more particularly the buttons. Before the advent of the synthetic buttons, this was the practice. The practice is still there in Mehsi area, but now the shells are to be imported from coastal towns. So there has been a lessening in the availability of this pearl button, this pearl Paresia, but the practice is still there. It is thriving. Next one. Then we must point out about the Gangetic dolphin, and then this Gangetic dolphin is the state aquatic animal, national aquatic, not a state, a national aquatic animal of India. It was declared by the then Prime Minister Manmohan Singh. Now about 50-kilometer stretch of the river Ganga, starting from this in Munger district. बोलो कौन सा है जगह जहाँ से पानी भरता है. So and again up to this Bikramshila. So 50-kilometer stretch. Sultanganj, from Sultanganj in Bhagalpur district to this Kahalgaon area in the site of the erstwhile University, Bikramshila University. So this 50-kilometer stretch is there as the Gangetic, as the dolphin sanctuary in Bihar. I think in India it is the first of its kind. And dolphin is a mammal, aquatic mammal, looks like fish. And Professor R.K. Sinha of Patna University, he is known as the Dolphin Man of Bihar. He was awarded Padma Shri for his work on this dolphin. And recently there was, they were dropped from one place to another place into the river Mahananda. So it made news. This is an example, the first ever rescue operation in Bihar of the Gangetic dolphin. Next one. Next one. Yes. So from this again, we are coming back to this. On the left side, we have Chenoplectiella articulata that is growing lavishly in the inundated fields. Then we have this Vetiveria zizanivoides, and then on the road side also we have this Vetiveria zizanivoides that provides strength to the earthen road that is there, and this is called Cas. So grows a species of sacrum that also grows abundantly during this time. Used for thatching purpose. Now here again we have on the right side we have Canna species and grows abundantly in the floodplains. Now this has proved its worth in the effluent treatment process. I had an occasion to go to Nimli, Rajasthan a few months back. Extensively used for effluent treatment, and here we have a makhana processing just instrument machine that is there. Next one. Now here we have examples of this Quax lacrima jobi in the middle, and Lenia coromandelica that is extensively used in the barebs, that is groves of this beetle. Here we have this simmer plant. The area has got a number of villages named as Simera, Simri, Simaria, etc. So this Salmelia malabaricum grows extensively in the floodplains. Next one. Now here again we are providing how a pond in which single Euryalphorax plant was there that developed about a dozen, I think, leaves. It was allowed unhindered space. That situation is not a normal situation, this situation, but at Makhana Research Institute in Darbhanga, one of our students, he made a photograph how this unfolding leaf then mature leaves, then decaying leaves. All these are there. And on the middle side, we have shown how the species of Nymphia called as bhaint, that is there, that is processed. And on the left side, we have white lotus that grows extensively in our area. Then we have the example from Makhana Research Center, how a spineless variety of this trapa is being grown there. And on the right side, we have the example how this makhana has got its ritualistic use. A bride in the first year of his marriage is offered makhana pops from the bride's side, and it is a practice on the day of Kojagra, that is Ashwin Purnima, and the use of makhana in Mithila ritual is a must on the occasion of Kojagra. So it is deeply intricated in our Mithila culture. This is a fine example how biodiversity, local ecodiversity is used and has become an integral component of the regional practices. Next one. Now here again, just best quality of makhana. 2 pops are there. Then the seeds, raw seeds, they have been stored there, and just see how just this one of a pond on which a plant has grown, probably through bird dispersal. Now this practice is there. Then on the left side, we have Rheomex dentatus plant that is called as banpalak. People use it as a green sag, leafy vegetable, and they provide fodder, used as fodder by the animals also. It grows lavishly alongside the rivers. It is a common practice here in this area. Next one. Just see how on the middle, this one, this is the appliance, bamboo appliance that is used by the local fishermen for harvesting of makhana from the ponds. Now then here we have the makhana seed coats that are simply used, that simply burnt in pop making. Now, this is not a good practice. A number of drug items now have been taken out from these seed coats. On the right side, you see how women, they are making the pops, just grading the pops into separate. This slide also shows the same. Then in the middle we have Ipomia aquatica, that is karmi sag, that is used more particularly on new entry into a newly constructed house. That is Grih Pravesh Day. It is considered very auspicious. It is highly nutritious, used extensively as leafy green by the local people, also fed to the animals. And on the right side, we have the flowers of this Nymphia species called bhaint or coca. Now that is be offered to the deity during Chaut Chandra Day in the Indian calendar month of this Bhadrapad. And here in the middle, the newspaper clipping is there, how women, they go for collection of fodder in the situation when there is water all around, and the male people, they just ferry the boat and they collect the fodder. So this is how the livelihood is there, dependent on the aquatic biodiversity in our area. Next one. So with this, I would like to end up here. And this is this Mithila painting is there. So the point that I would like to raise here is that we have no other option rather than the available phytodiversity, aquatic animal diversity that is used by the local people. And when there is no way out, people resort to these practices of using the native biodiversity for their various livelihood purposes, medicinal purposes, shelter purposes, housing purposes, whatever is possible, they are doing. So this is their lifestyle, and a fine example of using local biodiversity. I have tried my best to provide a glimpse of the situation that is there during this period. Thank you.
Thank you, sir. The forum is now open for discussion and doubts. Any questions? Okay. Our third resource person for today is Dr. Nirmali Gogoi, Assistant Professor, Department of Environmental Science, Tezpur University, Assam. On to you, ma'am.
दोनों ही म्यूट करो
Good evening. I want to thank the organizer for giving me the opportunity to participate in this important seminar webinar. So directly, as time is very precious, directly I will go to my presentation.
Madam, yes.
So my topic was biodiversity under changing climate threats and mitigation. So we all know about the global climate change, which is not new actually. This is a very common word nowadays. Everybody talk on it: climate change, global warming, and all. So we all are experiencing, in fact, the changing climate and all. And we know that human-induced changes in the climate is that a major cause than the natural cause. Nature is also responsible for little bit change in the climate, but that is very negligible. But human are mainly, responsible. And the climate change, it is giving so many changes. Temperature is rising, sea level is rising, the ocean is acidifying, and climate change is reflected in water cycle changes and in the extreme weather. So that all we are experiencing. Now, as for today's topic, my discussion is basically, how this changing climate, it is affecting the biodiversity. So we all know that climate and biodiversity has a very strong link, and that link actually, responsible. We cannot separate actually climate and biodiversity. We cannot separate. Any changes in any of the part is definitely going to, impact the other part. Climate change has a direct and indirect impact on biodiversity. At the same time, loss of biodiversity magnifies the adverse effect of climate change. And that is the major issue actually, the climate change we cannot, neglect with regard to the biodiversity, maintenance of the biodiversity. Biodiversity is threatened by climate change, but proper management of biodiversity can reduce the impact of climate change. That means, by protecting our biodiversity also, we can, manage the climate little bit. Okay. So this is the, diagram which is self-explanatory. So from where you can easily understand how, the climate change and biodiversity loss is linked. We know climate is changing, and this climate change has increasing the extreme events like floods, drought, and fires, and that is leading to the soil erosion. And soil erosion is causing, reduced primary production and nutrient cycling. Primary production is basically, the plant are responsible because they are the autotrophs. And as the primary production is reducing, so there is aggravation of the desertification. So desertification, that reduce the carbon sequestration into the soil. All the things, the carbon sequestration is reduced. And as the sequestration is reduced, definitely carbon will be released, as carbon dioxide gas. And that is again aggravating the climate change, climate change. And that climate change is again giving hindrance in the biodiversity, that aggravate the biodiversity loss. And this is how actually, both climate change and biodiversity are very much linked. Very much linked. And now the questions come as, climate is changing, that is a truth, and biodiversity is impacting because of the climate change. So we need to look at actually what are the, ecosystem, how ecosystem or the, species are experiencing this, change. So when we talk about the experience of the climate change at the ecosystem level or the species, and level. So basically it is due to the range shift, timing of biological activity, growth rate, relative abundance of species, cycling of water and nutrients, the risks of disturbance from fire, insect, and invasive species. So when I talk about the range shift, that basically talk about the species are relocating to areas with more tolerable climate conditions, because you see we human can and some animal can move, but the plant cannot move. But range shift, particularly, it threatens the species that cannot move fast enough. Okay? Range shift is a very common tactic under any harsh environment, so that everybody has the ability to protect or to thrive under a harsh environment, and for that they do they just follow any of the tactic on. And range shift, we can say it is one of the tactic like that. And but this is a kind of species which cannot move fast. For those species it is not possible, and they will be threatened more. Okay, depending on conditions that are becoming more rare. The depend on conditions that are becoming more rare. For those also it is difficult. And another issue is the timing of biological activity. We all know all plants, they have a certain time when they flower. Okay, and if you see in the nature or in the ecosystem, everything is linked in such a beautiful, manner, that the flower will bloom, and at the same time some insects will be available to to just take the honey from that flower, and like that everything is very much linked. And the time of the year is also much favorable to grow up the new generation of the plant or animal or insect. But timing of biological activity is changing due to the climate change. And we often see that some seasonal biological activities are happening 15 to 20 days earlier than several decades ago. Trees blooming earlier, migratory birds, they arriving earlier, butterflies, they emerging earlier. So though it looks like only 2 weeks or 3 weeks earlier, but it has a very very huge impact when we look at the ecosystem level, because ecosystem, in an ecosystem, the food chain, maintenance of the food chain is a very very important point. If something is not appearing in the food chain, that means the trophic level for that particular entity is going to be hampered in a huge way. And in a particular trophic level, if it is affected, definitely the upper trophic level will be affected in a very big manner. So that is how actually the timing, maintaining timing, biological timing is very very important, and that is disturbed actually by climate change. And that on the other way, it is affecting the biological diversity. It has a very very huge impact on the biological diversity, maintenance of the biological diversity. And changes in timing differ from species to species, so ecological interactions are disrupted. Maintenance of ecological interaction is very very important for a healthy biological diversity, for a strong biological diversity to maintain. Ecological interactions have to be maintained well. But this timing of biological activity, changes in the timing of biological activity, is not allowing it to maintain. And second thing is the biological biodiversity and climate change in various ecosystem. If we look at and polar ecosystem, basically, the polar ecosystem are home to an array of plant and animals that survive in some of the most extreme conditions, and which support a rich marine food chain. We look at the marine food chain. So this polar, ecosystem, it support it. But this ecosystem is, thus affected by the changing climate. Because it has an important role on the annual cycle of migratory birds. During 20th century, if you see Arctic air temperature, it is increased by approximately 5 degrees centigrade, and that is giving impact. That is impacted the polar ecosystem. So observed impacts are, seen in the form of livelihood of the indigenous people and polar bears, walrus, and the seals. These are some of the animals of that particular regions. They have been seen that these are impacted in a huge manner. So then another very important ecosystem which is linked with our food chain, that is the agricultural ecosystem, we can see that some extensive and varied impacts of climate change is seen. Increased exposure to heat stress, changes in rainfall pattern. We all experience that our rainfall pattern is changing, right? So we are experiencing a very, very erratic rainfall. That means there will be no rain for few months and there will be very heavy rain for 1 or 2 weeks continuously. So that is very much affecting the agricultural ecosystem, because the plants which are crops which are grown in between those drier months definitely suffered from drought. But the plants or the crops which fall under during these, heavy rain months, that will also affected due to flood. Okay, so greater leaching of nutrient, moreover, it aggravates the leaching of the nutrients from the soil during intense rain. When there is intense rain, so what will happen, the upper soil layer will be, runoff. There will be runoff water of the upper soil layer, and upper soil layer is the richest layer in terms of the nutrient and the fertility. And when it is lost, means the soil is losing all the nutrients, and plant will be suffer, crop will be suffer from nutrient stress. Okay? So also it is also aggravating the erosion. So due to strong wind and wildfires and drier regions, when soil is dry, if heavy rain, heavy wind comes, and even heavy rain comes, the soil will be eroded. So that leads to the reduced yield as much as 1/3 of the tropics and subtropics. So if you look at the dry and subhumid land ecosystem, which is actually very, very important ecosystem, they are the home to 35% of the global population. So deserts are projected to become hotter and drier, which would threaten the organisms that are already near their heat tolerance limit. Already these, the organisms are near the heat tolerance limit, and if 1 degree centigrade increase in temperature is also there, then that they are going to be impacted in a huge manner. Changes in rainfall pattern could also have serious impact on dry land biodiversity. Okay. So another very important ecosystem is the forest ecosystem. We all know forest is a, very, very important for maintenance of forest ecosystem is very, very important for climate, resilience. 1/3 of the earth's surface is covered by forest and are estimated to contain as much as 2/3 of all known terrestrial species. Forests could be threatened by pests and fires, making them more vulnerable to invasive species. So goods and services that forests provide would also be hampered. We all know that forest, it has many services forests are giving to us. Okay, so like that, inland water ecosystem is also going to be impacted under changing climate. More than 20% fish species are extinct, threatened and endangered in recent decades. So changing rainfall and melt pattern will affect the spawning and feeding habitat of many species. So these are some of the impact we are facing in the inland water ecosystem, starting from the coral bleaching, and that again impacting the tourism sector. That means economic loss also we are facing. Okay, so like that, mountain ecosystem is also affecting in a very large manner. It covers about 27% of the earth's surface and support 22% of the world's people. Climate change, it causes disappearance of alpine species, including the plants. Shrinking of glacier modifies the water holding capacities of mountains, thus affecting the downstream ecosystem. Okay, because in the downstream ecosystem, we need to depend upon the water that we get from the the rainfall which is there in the mountain, and they maintain a very beautiful linkage in the downstream and the upstream ecosystem. But the shrinking of the glacier, it just disrupt this linkage. So after all, these ecosystem, we can see that disturbance due to climate change, it causes loss in habitat. It causes loss in habitat, and that loss in habitat ultimately it causes extinction of the species. Species extinction is there, and loss in habitat not only just decrease the population, but species are endangered, and that some of the species even lost. And that loss in species, not only we are losing that particular species actually, but the beautiful food chain that they are maintaining in that particular ecosystem, that is also going to be disturbed. And that disturbance in the food chain is ultimately going to give pressure in the upper trophic level. Suppose in the lower trophic level we lost a species, but upper trophic level they are all right, we can think, but that is they are not all right, right? Because they will be losing their own food. Okay, so that is the reason why we have to think about that climate change impacts on the biodiversity. So these are some of the examples of how these species, they are all are affected due to the changing climate. I will not go in detail this, you can read it out. So warmer temperature in Pacific regions, they reduce the number of male sea turtle. So male, if it is the not only the species loss, sometimes it is also seen that the loss in species, it is some sex wise, means sometimes the male species are more affected, sometimes the female species are more affected, but ultimately their population is going to be impacted, right? So this is how actually diverse way they ultimately going to impact the species loss. Okay, the climate change and food security, if we look at the food security under changing climatic scenario, so we see that direct impacts on plant and animal growth and food processing and distribution network is the major cause. And indirectly, we can see that social, economic, and political structure. So, for example, higher temperature, it causes longer growing season and increased productivity of high productivity in high latitude, as long as temperature increases do not exceed 3 degrees centigrade. Crop losses and declines in productivity in low latitude. Less winter kill but increased thermal stress for livestock. Earlier spring activity, greater winter survival, and expansion of ranges. Okay, these are some of the causes which ultimately leads to the leads to the threatening the food security. Okay, climate change and food security is, it is due to the precipitation changes in precipitation, reduction in arable land in areas with decreased precipitation, loss available of water for crops, less availability of water for crops, declining aquaculture in areas of decreased precipitation. So the crop failure is there. So now, if we look at the forest, forests are also, they have a great impact on the carbon shrinking the carbon from the atmosphere. They act as a sink to for the atmospheric carbon, and the global forest sector emission from deforestation, it releases about 1.6 gigaton of carbon per year, so which is equivalent to 20% of anthropogenic greenhouse gases. Okay, and also forests, they affect and are affected by climate change. Outcome determines mitigation, adaptation, and an adaptation potential. Outcome that determines the mitigation and adaptation potential, that will determine how it will work as a sink for carbon. So how the forest affect as a carbon cycle, that that is how it reduce the emission and it increase as a sink. That means we know that the carbon from the atmosphere, it is in the carbon dioxide gas, and the forest during their process of photosynthesis, it take up the carbon dioxide and it's stored in the body, its body. But you can also ask that how, because the any plant can do that, why forest? Because in the forest actually what happened, the trees are mostly perennial in nature, right? But which is not there in the crop plant. Crop plants, most of them are very much seasonal, right? Very few crop plants are there, like rubber, tea, like that, which is perennial in nature. But if it is seasonal, definitely the carbon which is stored in the body of the plant, that is again after cutting down, it is again going to be mixed in the soil, and during that process of decomposition, it is again going to release the carbon dioxide, and which normally not happen in the forest. And moreover, the forest also it maintains a very beautiful ecosystem that is not disturbed actually. But agriculture is a managed ecosystem, but forest is a natural ecosystem. So the beautiful linkage which we get in the forest, that helps, that support in the water hydrological cycle to maintain, nutrient cycling to maintain in a very beautiful way. Okay, so that is not there in case of managed ecosystem. So invasive species and climate change, when we look at some of the invasive species are getting chance with the changes in the climate. If you see the biodiversity, some invasive species are coming up in each of the ecosystem, and that invasive species are creating so much havoc. Okay, so these are some of the invasive species. For example, mountain pine beetle are one of that invasive species. That due to increased temperature, this species is found to be there in some of the warmer winter areas. They are causing a problem in those ecosystem. And another is the kudzu. Kudzu introduces into US. So, but for soil erosion, actually, basically it was used, earlier, but it has become a rampant weed in the southwest US. So, the invasive species, definitely it will be a problem when it enters in any ecosystem. Like that, cactus moth is another, very very notorious invasive species in Caribbean. So like that, these invasive species are creating problem in the biodiversity of any ecosystem where actually it entered. So now the questions come, how to mitigate actually, how to mitigate. Already we have seen that climate change is creating problem in the biodiversity. So how to mitigate? So when we talk about the mitigation measures, so more particularly, first of all, if we talk about the agricultural ecosystem, whatever biodiversity changes it is taking place due to climate change in the agricultural ecosystem, how we can get a resilient agricultural system. So for that, the drought tolerant species help resistance to climate change and diseases. Better practices generate healthier soils and may help carbon dioxide sequestration and water storage. Selective irrigation, mulching, cover crops, crop rotation, reduce plowing, and agroforestry can help in this regard. Changing agricultural practices, there are several measures to reduce carbon emissions from agriculture which target both carbon dioxide and methane. Okay, so they include zero tillage. Tillage is very important practice in agriculture, but it is found by the scientists that when we go for a very heavy tillage, it has many environmental, many adverse effect in the environment. So zero tillage means that we will till the area where we are going to put the sapling or put the seed, not the whole area. that is the zero tillage, minimum tillage, and polyculture, crop residue, new strains of plants, managing the manure, because otherwise what happen if you allow the manure to just decay anyhow, so that is going to produce many of the greenhouse gases like methane, nitrous oxide. But if we just manage the manure in a scientific way, that can be very very helpful for the environmental, because manure otherwise very very rich in nutrients. Okay, so mitigation measures, for example, in case of wetland restoration is important practice in this regard. Trans-government initiatives such as International Convention on Wetlands (Ramsar) and the European Union Habitats directives promote restoration projects around the world. So you see, in this case, land use planning is very very important. This is kind of some land use zoning, building restrictions in vulnerable floodplains on low-lying coast is very very important. Enforcing strict runoff control by this type of structure, soakways. Soakways, these are some of the dry, dry storage areas where we can store the, those waste in these structures. So solar radiation management, that is SRM, is another important thing, which is a new coming up in some of the developed countries. Actually, some plan they are making so that we can reduce the climate change impact. Geoengineering involvements are there, so deliberately interventing the climate system to counteract global warming. One proposal is to use orbiting the satellite to reflect some inward radiation back into space, rather like a giant sunset. So this type of structures they are proposing, so that we can just reduce the global warming, right? This could cool the earth within months and be relatively cheap compared with mitigation. This is some of the engineering techniques that is proposed actually, so that we can reduce the global warming. So apart from that, some of the international agreements we all know, Kyoto Protocol is there. So the Paris Climate Convention is there. Kyoto Protocol, many developed countries they agreed to legally binding reductions in their carbon dioxide emissions. The aim was to bring about 5% cut in global GHG emissions from 1990 levels by 2008-2012. Okay, and Paris Climate Convention COP21 ended with an agreement to reduce global carbon dioxide emissions to below 60% up to 2010 level, and to restrict global warming to 2 degrees centigrade increase by 2050. So apart from that, countries also set their own voluntary targets, and there is no detailed timetable. So it has been agreed that developed countries will transfer funds and technologies to help developing countries to achieve their targets. I guess you might be knowing about this, carbon credit, right? So this is nothing but the carbon credit. Actually, the credit can be bought. If suppose a country is a more polluter country, so they emit more carbon dioxide, they can buy carbon credit from another developed countries by giving some money, right? But that country who is selling the carbon credit, they must have to develop some eco-friendly techniques. Suppose they have to go for plantation or some other, environment friendly technique they have to develop to just counterbalance the, emission that has been made by the other country. Okay, that is called carbon credit. It is now very very important hot market. Actually, there is a big huge carbon market is going on among the developed and developing countries. So the current relationship between the big energy providers and the government dictates the amount of switching from fossil fuel to renewables and nuclear power. Renewables, that means solar, wind, and wave provide intermittent electricity, while fossil fuel and nuclear provide the continuous power essential for our current infrastructure. So, but so everything is in place, but we know that human are responsible, right? So we are responsible actually. If we now whatever I have said about the mitigation, you see everything is in place. Technological development is also going on. We all know that the fossil fuel burning of the fossil fuel is responsible for climate change, and which is affecting the biodiversity also. But fossil fuel is replaced now in that most of the developed countries, and even developing country like us also, we are phasing out the fossil fuel now. Government is also giving intensive incentive to the for if someone wants to buy solar panels. Even my university is having that solar energy we are using. Almost 30% is solar, and most of the buildings are solar. And also government is now introducing the e-bike, e-cars and all, giving emphasis on those sectors. Like that, we have all those international convention, as I have already mentioned, in place. And also other techniques scientifically, we also know the know-how, scientific know-how is also with us. But only the problem is the our rule, individual rule. We have to play a very, very strong positive role to overcome this particular issue, that climate change. Then only it will be possible. The our own rule is very, very important in this regard. So our outlook have to be changed. Okay, changes in activities at the personal, community, and national level can affect the rate of future climate change and species abilities to adapt. Okay, that means the biodiversity. Some of the areas where changes in human activities could help species adapt include approaches to agriculture, water management practices, energy source and use, transportation, pollution remediation, biological conservation, and much more. You see, I strongly believe, in my level, I strongly believe for all these act actually, we also have some laws in place in India. But I don't think that for those act, if through law we can do something, right? At personal level, that have to come from our mind, right? Like that, we believe in God. We have some religious values. Personally, everybody has some different religious values. So like that, the earth is the mother earth, and we have some value for her, and we have to change our outlook, and then only it will be possible that we can normalize the climate, and that normalization in climate ultimately can give us a good biodiversity in the mother in our art. Okay, that's all. Thank you so much for listening.
Thank you so much, ma'am. The forum is open for discussion.
Yeah.
Any questions?
I guess I was very, fast. I don't know. I thought there is a timing, thing. Oh, that's why I go too fast. If somebody wants to ask anything, I am open to, just give my number here. So anybody can call me even if you just like in future also.
Okay, ma'am. Thank you so much, ma'am.
Thank you.
Hello. Our last and final resource person for today is Dr. D. Tangamani, Scientist, FGRM Division, IFG and DB, Coimbatore, Tamil Nadu. On to you, ma'am.
Good evening, yes. My slide is visible?
Yes, madam, yes.
Good evening, all the learned participants and youngsters. Myself, Dr. Tangamani from Institute of Forest Genetics and Tree Breeding, and I would like to share with you regarding the forest genetic resources of India and its utilization for our future. And before going for this one, I want to clarify what is forest genetic resources. Forest genetic resources, it simply refers to heritable materials, that is genes, that is DNA, which is present in a tree and other woody species that should be useful for economically, environmentally, scientifically, and societally. So this word FGR has been coined by FAO in the year 2014. So they have given a term only for forest genetic resources and for its conservation in the year 2004. And they have started this one. And why we have to do this this forest genetic resource conservation characterization means we know we have to conserve our earth resources for our sustainable utilization for human survival. And because of the pressure of increased urbanization and because of natural calamities, etc. So there is a destroying of forest. Nearly 17 million hectares of forest has been destroyed every year. So we have to conserve all our resources. So for that we need to conserve this forest genetic resources. So we should know the variations, differences between the biodiversity conservation and forest genetic resources conservation, right? So we all know that biodiversity, about the biodiversity conservation, that is flora, fauna of all the living organisms of that is the biodiversity. But in FGR it concerns only about the trees and the woody species here. And in biodiversity, and we feel that rare and endangered species, that's very more important, so we are conserving that one. But in FGR, potentially important species which are useful for different industries, different stakeholders, we are conserving. And in biodiversity, primarily it is conserved in its original ecosystem, wherever it is, in natural forest or ecosystem, like that. And it is maintained mainly in wild, and the entire spectrum of variability is maintained as such in the in situ conservation in the same area. But in FGR, in forest genetic resources, it is primarily conserved in ex situ, that is as in seed bank, in field gene bank, like that. And from the nature we are collecting and we are initiating domestication for its utilization, and its variability is studied and selected for its trading, economic trend, and we are domesticating and using for different industries. And what is happening internationally in forest genetic resources management, we should know, because not only in India and in FAO, so they are preparing and updating state of world forest genetic resources, and India is one of the member, and it has been included in the year 2012, and they have prepared we have prepared the survey FGR report in the year 2014, and we are doing the regular discussions, and regularly every year we are updating all our about our resources and what all the work has been carried out. All those things we are updating with them. So this upper forgen, that is Asia Pacific Forest Genetic Resource Program, which has been initiated in the year 2003 with 14 participating countries, including India. So that is the worldwide how this FGRM program is carried out, and we prioritize the species across India. We prioritize the woody species. Nearly 272 species we have prioritized. It is based upon the environmental and societal values, and it is considered as threatened, whether it is threatened and what status, and based on this genetic variability, all those things we are prioritized. And what all the work we have done up to, we have evaluated up to 104, and we considered 261 species of threatened, and we have given importance of environmental and social values of 195. All these things we have done in India, and in peninsular India, based upon the meeting with the foresters and stakeholders, and with the IFS Indian Forest Officers, and different forest oriented industries, we have prioritized, we prioritized the species like teak, melena, casuarina, eucalyptus, Pterocarpus marxum, acacias, sapindus, bale. So all these species, based on its economic value and potentiality, we have prioritized. So all these resources we have to explore it from different locations, and we have to assemble in one area as a gene bank or germplasm bank. We have to find out the variability between them, and we have to domesticate the very good one. We have to rank all the germplasm, and the very good one which is showing high variability, we are mass multiplying, and we are giving it for farmers for tree improvement program. So this MGR Forest Genetic Resource Management Program, it is a constitutive following activities. It is mainly exploration and collection from the natural populations, and characterization, evaluation and documentation, and conservation and regeneration, and it has having a germplasm exchange and supply of user agencies and plant quarantine. So these are the important components, and from where and all we are taking the genetic resources from the naturalized forms, from the reserve forest and natural forest, or from the land races, and from the genetic stocks, and from the released clones, the parental lines and hybrids, and from the farmer selections also we are taking the genetic materials. So what all these components, all these materials we are taking, and we are finding the network between the different agencies, that is farmers, stakeholders and foresters, etc., and we are finding the variations, heritable variations between the species and between the regions and populations, and we are managing them in situ conservation as well as in ex situ conservation. In situ conservation, in the sense it is conserved in the means of biosphere reserve, national parks, sanctuaries, preservation plots, and seed sands, sacred groves, and community reserves. In these ways we are conserving. And in ex situ conservations, we are conserving in seed gene bank, in vitro gene bank, and cryogene bank, seed orchards, and clonal repositories, in arboretums, in plantations, and as a herbal gardens we are maintaining, as a botanical garden also we are maintaining. And we are conserving all these things. And what is characterization means? We are selecting the germplasms from different area, and for example, for teak, we are taking from entire India, from Andhra Pradesh, from Tamil Nadu, from Karnataka, etc., and all are take only, but we have to characterize them. So we have to take a passport data of that particular tree, plant or organism. So it's a full description about that, how it look morphologically, and what are the biochemical components in it, how it varies, and how it varies molecularly. So within, between, and among the populations, all those things we are doing characterizing, and from the different, from 10 or 20 or 100 or 600, we are narrowing down the germplasms based upon its high variations for our tree improvement program. So for this narrowing down, which is bringing the genetic characters, the germplasm which express more characters. So those type of germplasms we need for our tree improvement program for industrial utilization and domestications. So for that, for that we are exploring, so we are finding the variations. So this finding variations is very much important process for evolution, and because due to change in this genetic makeup of an organisms, this sort of variation occurs. So whether this is a valid variations or not, all those things we have to explore it. So this variation only, it passes one generation to next generations. So this variation occurs also occurs because of heredity. Also this occurs because of the environment, edaphic factors and climatic factors, right? So these are all the variants we know in agriculture and in forestry, in everywhere. Daily in the market we are seeing this variants, right? These are all the coconut which is collected from Lakshadweep. See the variability. All are Cocos nucifera only. See the variations. Similarly, see the citrus diversity in Northeast India itself. See the variations in the same locations. We could see the different size, flavor, and taste, and the growth morphology also. It differs. A different diversity of citrus we could find, and similarly Hemidesmus indicus. So see the variations in the leaf morphology itself. So not only first we are collecting based on its morphological variations. Later we are characterizing using biochemical methods as well as molecular methods, and we are exploring to the stakeholders. So selection of the species from the nature for their conservations, it is mainly we choose based on economic importance or environmental importance or ecological importance or societal importance. So and based upon the threatened status of the species also very much important. In what status it is vulnerable, extinct stage, or we have to so all those status it is very much important. And next one is in what way we are conserving this species, whether it's in situ or in germplasm bank or seed banks. All these criteria are very much important for selection of the species and conservation of the species. So in our research studies, we know we are working with the teak, Tectona grandis. We know teak is the wealth of India. It has great variations in its roots because of its height, diameter, flowering age, stem form, and branching pattern, wood color, and wood extractives, etc. So we have explored different areas, different teaks from its natural populations as well as from the plantations in different areas of Western Ghats. And first we are seeing this crown shape and branching pattern, how it looks. So it's a primary one, primary observations, this branching patterns and the crown shape. Later, we are visualizing we are feeling the presence or absence of any biochemical or anything. We are visualizing. So anthocyanin, in case of teak, it is anthocyanin, whether it is present or not in medium or strong, like that we are assessing, and we are seeing the variations in leaf morphology. And we are seeing its petiole length and leaf morphology, whether it is a sessile or a petiolate, like that, and the length and all we are measuring, and we are seeing this various petiole length, and we are seeing its shape, its tips, what type of apex shape. And we are seeing the texture of the leaf, whether it's a glabrous, leathery, scabrous, like that. And we are seeing its hairs, trichomes in upper dorsal face and then lower face, in how it is, the hair arrangement is, whether high or medium, and what size, all those things. And we are seeing the leaf arrangements also, how it is same teak only, how it is opposite or decussate or ternate, like that. And similarly, we are observing its flower. We observe this flower petal color and anthers and the petal numbers, anthers numbers, etc. And similarly, we observed the cortex, the fruit exocarp also, whether it is tight or loose or sharp or round, like that. And we are documenting everything. We are mapping. We are documenting. We are mapping everything wherever it is. We went and we documenting everything. using different phases, and we are finalizing and seeing all the phenophases of teak, when it flower, when it is leafless, when it flowers, when it fruits, or when it ripens, everything, and regeneration studies, everything we are recording. And whole year, when it flowers and when leaf flush, and all the phenologies for this natural populations as well as in plantations, we observed, and we are seeing the variations. And and all those things, flowering initiation up to fruit initiation, ripen fruits, all those things we are recording. And the phenology studies of the teak also, natural populations also we are recording. Up to 25% flower bloom, up to 60% percentage, up to 80%, and full flower bloom, fruit initiation, all those things we are recording, and we are seeing the variations. And similarly, we are sampled in all the plantation as well as in natural, and with more than 400 centimeter girth also we have recorded in the Western Ghats. So we through SR markers, through next generation sequencing, using SR simple sequence repeats microsatellite markers, we have explored several genes, which is very much important for durability of the timber. So we explored all those the genes. While doing through different softwares, we found out this genetic diversity in teak populations in different areas, different states of Kerala, Andhra Pradesh, Karnataka, and Tamil Nadu, and from where it has been evolved, from where and all it is disseminated. All those things we have recorded. And similarly, in rosewood, Dalbergia latifolia, similar type of exploration and characterization and conservation work we have done in the rosewood from Western Ghats in Kerala area and in Tamil Nadu area. We have collected different, more than 57 germplasms we have recorded, we have collected, and the root cuttings were taken, and the sprouts were grown in the nursery, and they were very well propagated and hardened, and they were planted in the germplasm bank. We are having 3 different areas. We are maintaining this field gene bank. So for this one, whatever the germplasm we collected across Western Ghats, we do morphological characterization, and through this image analysis from the field observations as well as from the lab observations, so using the image analyzer equipment and this softwares, we are seeing the variations, how it differs in different climatic conditions, and first leaf, second leaf, third leaf, like that. We are seeing the morphological variations. And similarly, the primary and secondary metabolites in different climatic conditions in different areas, edaphic conditions, we are quantifying all the secondary metabolites and primary metabolites. All those things we are tabulating, and we're using different softwares. We are finding out the variations through thin layer chromatography. This is a profile of this tannin, hexane extract, how it is showing the various chemical. So the germplasm is showing a high biochemical profile. We are selecting for further our tree improvement program. So similarly, the linkage between the different groups, between the different regions, we are finding out through different dendrogram constructions, and the molecular variations between and among the populations and individuals, we are finding out through the dominant marker like RAPD marker, as well as the codominant marker like ISR, SSRs, and SNPs. And through this SSR and SNPs, we could find this variations, high variations between the regions, between the individuals, between the populations, and the point mutations. This because of what this exact character, that's also we could we found out using SNP, next generation's Illumina sequencing technique. So we formulated this dendrogram also, which one has evolved from which region, it has been disseminated to what are the areas it has been disseminated, who's the how is the evolution process, and how much percentage the variations occurs among the regions, among the populations, and within the populations we have recorded in Dalbergia latifolia as well as in Dalbergia sessoides. And similarly, the different type of barks and teaks, see, all are Tectona grandis only. See the variations: scaly bark, smooth bark, and the gray color, fissured bark. All those things we have recorded. Similarly, in different species we are working. Okay, so these are some of the in eucalyptus also. See the variations: light brown, dark brown, gray color. All these variations we are recording. And similar type of work, FGR work we have done in Pomegme pinnata also. All morphological, biochemical and molecular work we have done, and we assembled all the germplasm in germplasm bank, and we ranked the species for its oil content. The high oil yielding and high fruit yielding germplasms were recorded, and they were mass multiplied, and we kept it. We have established a progeny trial and plantations in different areas of Tamil Nadu. And similarly, some of the important medicinal plants also we are working. See the diversity of Apis pictorius, see the seeds, different colors, the variations. And similarly, we are working with Enteta scandens. That's one of the important medicinal plants. In Eastern Ghats, we have collected, we conserved, and we in Koli Hills itself, we propagated, we characterized all the biochemicals and in this one, and so we assembled all the germplasms in germplasm bank. And we do the cytoconservations in cold storage in mid-term, long-term seeds in seed gene bank, and we do have cryo gene bank in -196 degrees centigrade using liquid nitrogen, and we could store the germplasms in tissue culture materials in intro gene bank also. And in as I told, we have several field stations, and we are having field gene bank. So different varieties of very varied sweet tamarind we do have, we assembled, and similarly different varied hazard rating content and sheet shapes and growth pattern. Neem we have assembled, and similarly teak orchard, seedling seed orchard, clonal seed orchards, and we are having, and for Pomegme peanut also we are having, and we are having different populations of Castrona equistifolia, Zumniana, and Ailanthus excelsa species, and we do the trial, provenance trial, and trial in different areas for Tamil Nadu and Andhra and Karnataka. And we need we are seeking the help of this forest departments for this conservation, forest and resource conservation explorations and characterization, because funding is very much important for different studies of this priority species, and for strengthening the local community for conservation of this forest and resource species through participatory approaches. We need the support of them, and for managing all those lands of permanent preservation plots, all the areas, we need the support of the forest department. So, and we have to control the encroachment and the invasive species attack also. So all the things can be done only by the forest departments. And we observed that there is no single method to conserve all the genetic diversity within a species. So complementary approach is required. So conservation in natural population of collections in gene bank. So appropriate strategy, it depends upon biological characterization of species and its infrastructure and available resources, and a geographic site, and understanding of extent and distribution of genetic diversity. So network collaboration with the different stakeholders, forest department and farmers is very much necessary for conservation of the forest genetic resources. So youngsters, all the youngsters and natural lovers, they should know about the variation among the plants, and they should conserve them. We should not neglect any single plantlets, any single trees because of its stunt growth, because of its obnoxious thing. We should not cull it out. We have to conserve all these things for its for our futures, because genetic variability is a very much important one. We know it is because of a genotype, it may be because of the environment, but this is this is very much important for our generations. We can explore all these things using our different biochemical markers and molecular markers. Okay, so thank you very much for this wonderful opportunity to share about my work in forest genetic resources and conservation characterization. Thank you, organizers.
Thank you, ma'am. The forum is now open for discussion and doubts. Any questions? Okay. Now we move on to the technical session 2 of this event, and the rapporteur for the same is Dr. Pampi Ghosh, HOD, Department of Botany, Seva Bharati Mahavidyalay, Kabghari, Jhargram, West Bengal. On to you, ma'am.
Thank you, Meena. Now we are going to start our technical session 2, and here 13 presenters. In this session, today's chairperson is Professor R. Bhattacharya, retired professor, Presidency University, Kolkata. Then subject experts are Dr. Ghayal Nivedita A, Professor of Botany, Abhasaheb Gadawade College, Pune, MS. Then Dr. Vinuta R. Bhatta, Assistant Professor, Department of Zoology and Genetics, Jyoti Nivas College, Autonomous, Kodamangala, Bangalore. Then Dr. Soma Pal Saha, Associate Professor, Microbiology, Lady Brabourne College, Kolkata. Then Dr. Nateshan Payer Nadimuthu, Assistant Professor, Botany, Tagore Government Arts and Science College, Puducherry. So, with this, I would like to request our today's chairperson, Professor R. Bhattacharya, and other experts to run the session, and please strictly follow time, and it is 5 minutes for each presenters. Sir.
Thank you, madam.
Yes.
Thank you, madam. I
Our
welcome
first presenter is Shreya Roy. Sir, yes, sir.
Shreya,
Shreya Roy.
Shreya, please start your lecture.
Shreya? 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 Shreya is not here. So, sir, next presenter is Maria Dev Burma.
Now, anybody other of that group, they will present?
Umme Hasanat. I think she is also not.
No, Shreya is in the meeting. Shreya is in the meeting, we can see.
Shreya is meeting. Then Shreya?
Yes, ma'am. No, ma'am. I'm Umme Hasanat.
Then Umme Hasanat also is here, and her presentation on the 4th. So, Umme, you can start your presentation.
Okay.
Hello, ma'am.
Okay.
Madam, I am present. Maria de Burma.
First, Maria de Burma, then sir, first decide. Umme at 4th position and Maria in the 2nd.
Okay.
Who will do? Sir.
So, who is going to present now, ma'am? Ume.
I have no problem to present, ma'am.
Ma'am, I am Shreya. I have no problem to present in first.
Then present. Why are you late?
Shreya, we are telling repeatedly. Shreya, you please start your presentation.
Yes, sir. I have some technical issue. I am presenting.
It's already late, so don't.
Shreya. Yes. Yes, ma'am. I'm sharing my.
Do you have any kind of problem?
Ma'am, everything okay?
Okay, start.
Yes, you start.
Yeah, start.
Yes, ma'am. What is it? Good evening, everyone. My today's topic is synergistic activity of tea with some common medicinal plant review. After water, tea is one of the most ancient, cheapest, and most widely consumed health beverage throughout the world for its health and sensory and stimulating and relaxing properties. Primary studies showed that the presence of secondary metabolites and presence of antioxidant and antimicrobial properties is present in tea, both as well as green tea and black tea. Tea having polyphenol flavonoids, especially catechins, are the leading functional components, which accounts for the 30% of its weight of green tea leaves. To enhance the tea flavor and medicinal properties, various tea combinations are widely available in market. The combination of plant products may exhibit different types of interaction like synergistic effect among their different phytochemicals. It may change or enhance their biological properties. Those varieties are absolute paramount, but limited scientific data is available about this. The synergistic effect is the effect of two or more process interacting together to produce an effect that is greater than the cumulative effect of those process produced when used individually. In medicine, we can say that the interaction of two or more drugs when their combined effect is greater than the effect seen when the drug is given alone. Objective of present review: the study of synergistic activity of green tea extract with some specific medicinal plant and comparison between the changes of phytochemicals content of green tea and black tea after adding some common medicinal plants. The green tea and black tea and both are with medicinal plants. Now to observe which one shows the better antioxidant activity and phytochemical contents. And this is a list of black tea and green tea and the phytochemicals present between them. Now this is a list of medicinal plants used with tea to enhance its property. First, ginger, tulsi, lemon, and clove. And ginger, we use the rhizome part, and tulsi's leaf, and lemon fruit part, and cloves flower part, and the piper nigram's fruit part. And this is a table of different phytochemicals present in the medicinal plants. Now coming to briefly discussing about the medicinal plant. Ginger root or ginger rhizome is widely used as spice and a folk medicine. The active principle gingerols and their corresponding dehydrating products are responsible for strong antioxidant activity. And black pepper is a popular spice, shows the potential antioxidant activity. The main bioactive constituent is piperine. It increases the bioavailability of many phytochemicals present in food items. Tulsi or osimum, it is commonly known as queen of plants. It is a very good source of eugenol and etc. For the presence of eugenol, it shows the antioxidant property, and also thought that it is responsible for the inhibition of lipid peroxidation. Lemon juice is content phenolic acid, ascorbic acid, citric acid. Lemonades are prevents many disease associated with oxidative stress. And clove, the main active components use eugenol, and it shows the antimicrobial and antioxidant activity. Now coming to discussion. In the present study, green tea and black tea used as a base. The individual medicinal plants and tea, ginger, black pepper, and tulsi, and different mixture of extract of tea with other medicinal plants have been evaluated for synergistic and anti-synergistic protein. Yeah, since the antioxidant potential of herbal extract mainly depends on the concentration of total polyphenol and flavonoids, and therefore we determine their concentration based on the standard method or with their free radical scavenging activity. And here, and according to the table, we can see the when the tea is mixed with tulsi, it show the better antioxidant activity. But when tea is mixed with ginger, black pepper, tulsi in 1:1:1 ratio, and it show the better excellent antioxidant content. Now tea is mixed with clove. In the present study, we come to know that the antioxidant and antimicrobial activity of the different clove-based herbal tea and the total polyphenol content and the total flavonoid content of green tea and black tea were determined and compared with the mixture of clove and tea respectively. Now first, green tea mixed with clove mixture and high concentration.
Yeah, please try to round up.
Hello?
It is already 6 minutes.
Sir, I'm only 2 minutes needed. When the high concentration of green tea and high concentration of clove mixture always show the better antioxidant activity, and in case of black tea, the low concentration of black tea and high concentration of clove mixture will show the better antioxidant activity according to the high concentration of black tea and the low concentration of clove mixture. Now tea with lemon. It was reported that the green tea has a high radical scavenging activity and DP and free radicals method. And the ability of lemon to chelate iron was weak, and when the tea is mixed with lemon, it always show the better antioxidant activity according to green tea. Now conclusion. On the basis of the above discussion, the antioxidant potential of the plant extract depend on the phenolic compounds and flavonoid present in the particular extract. Green tea osmum combination always show the better result, but when tea is mixed with accordingly with others, and it shows the better result. Thus, the study provides that the scientific basis infusion at the particular ratio at the maximum antioxidant potential. Due to the presence of additional phenolic compound in lemon, green tea with lemon also shows the better antioxidant activity. And further studies are needed using different tea brands with different supplementation others. Thus, it can be concluded that the selected extract combination produce their effect in the synergistic manner with green tea, and there is a scope for confirming the result of the study on relevant animal models, followed by the study for clinical support in future for development polyherbal formulation of green tea and black tea with a respective plant extract. And there are some differences. Thank you.
Thank you, sir. Any question that will be communicated through her email. So next speaker, Maria Dev Verma, please.
Thank you, sir.
Yes, sir. Okay.
Please try to maintain the time.
Yes, sir. Yes, sir. I'm trying to share my screen now. Okay, good evening, everyone. Today I'm going to present a review paper on potential of freshwater macrophytes for phytoremediation of wastewater. So in introduction, water pollution has become one of the serious ecological issues in the world over the last few years. Elimination of noxious pollutants is very much important to curtail the threats to the human health and ecosystems. So the proper treatment of wastewater is a method of environmental management that aims to avoid any kind of water pollutants and reduce the organic loads and recover the nutrients. There are many conventional wastewater treatment technologies which are uneconomical, complicated to operation, and time consuming. Therefore, these treatment technologies require high capital investment and ended with a problem of generation of sludge disposal. So to overcome these kind of problems and issues, we need to opt for an alternative techniques for treatment of wastewater which should be suitable and sustainable. Among various strategies developed so far, the techniques of phytoremediation using aquatic macrophyte is the most preferable technology to remove the pollutant from the wastewaters. Phytoremediation has been described as an advanced bioremediation method, utilizes plants and associated microorganisms to clean up the environmental pollutants. Therefore, the present study precises the potential application of freshwater macrophytes in phytoremediation for the treatment of the wastewater. The methodologies, as is a review paper, so these are the few journals from where I have collected my materials. Phytoremediation. This phytoremediation is the use of the plant techniques to remove or degrade the organic or inorganic matters constituent present in the soils and water. As shown in the figure, it is a process by different mechanisms, namely phytofiltration, phytostabilization, phytodegradation, and phytoextraction. So phytoremediation is one of the emerging technology over other conventional methods for the removals of pollutants from the wastewater and from the environment. So several techniques have been earlier used, but this is one of the cost-effective as well as environmentally friendly method to clean up the environment. So this method comprises operation like plant metal uptake, translocation, accumulation, and degradation. And microphytes and the phytoremediation. So aquatic plants act as a natural absorber for the contaminants and heavy metals. Therefore, phytoremediation is a promising green technology for moderate to low level of contamination. This clean-up technology using the aquatic macrophytes is aesthetically pleasing, energy efficient, cost-effective with a long-lasting applicability. Therefore, in the last few decades, aquatic plants have gained an overwhelming reputation because of their capacity to clean up the contaminant site throughout the world. The growth and cultivation of these aquatic plants are time-consuming, which may restrict the growing demand of the phytoremediation. Now, the types of aquatic microphytes which are used for the phytoremediation technology. The different aquatic plants are used successfully in the phytoremediation of the wastewater. The selection of aquatic plant species for accumulation of heavy metals is a very important matter to enhance the phytoremediation. And among the various aquatic microphytes, free-floating plants such as a member of Laminaceae like Lamina minor, Pistis stertotis, and the iconic recipes are those from the genera Salvinia have mostly preferred as a hyper-accumulator with high biomass, showing their potential in removal of heavy metals from the aqueous environment. Now, the few aquatic macrophytes used for water and wastewater treatment. From the floating aquatic macrophytes, the mostly used are the Cornea, Lamina, Pistia, and the Salvinia. So, floating aquatic macrophytes are with the submerged roots and floating leaves. Now, these floating macrophytes are highly suitable to eliminate the contaminants from the aquatic ecosystem due to their high productivity, high nutritive values, and easy harvesting process. Water hyacinth is Iconiac recipes. Water hyacinth is a free-floating aquatic plant which belongs to family Potendriaceae that is closely correlated with the lily family. It has an advanced tendency to remediate different pollutants like organic materials, heavy metals, total suspended solids, total dissolved solids, and the nutrients. This water hyacinth is recommended to treat industrial wastewater, domestic wastewater, stress effluent, and sludge ponds because it has high absorption rate of organic and inorganic contaminants, and it can tolerate an extremely polluted environment such as gigantic production rate of biomass. This species has greatly ability to remediate contaminants like arsenic, zinc, mercury, nickel, copper, and lead from the industrial and other domestic wastewater streams. The next species is the water lettuce, and the botanical name is Pistia statutes. This this is commonly known as a water lettuce and is feed in the aram family. This is Aracaceae. Water cabbage or Nile cabbage or water lettuce jalcumbi are the common name of this Pistia statues. It is mostly found in the lake, stream, and ponds. And the Pistia species have 20 centimeter wide to 10 to 20 centimeter long pale green leaves. This plant has the capacity of removing nutrients such as the BOD, COD, dissolved oxygen, pH, and then the total nitrogen, ammonia, nitrite, nitrate, phosphate from the drinking and the surface water, storm water, sewage water, and the industrial water. This plant also can uptake heavy metals like copper, zinc, iron, chromium, and cadmium. The next species which mostly used for the phytoremediation method is like water fan. This is Salvinia. Salvinia adana, small free-floating macrophytes extensively scattered in the aquatic ecosystem. Salvinia for use as a bioindicator pollutant index and for the phytoremediation as well. And Salvinia minima is successfully used for the remediation of highly strength synthetic organic wastewater. And different species of Salvinia are an excellent accumulator of iron, cadmium, nickel, manganese, zinc, and lead, as reported by several studies. Now, from the submerged macrophyte, aquatic macrophytes, the mostly used species are the Mycophyllum specticum, Ceratophyllum demersum, and the Potamegaton species, and the Velicenaria. So these are the few submerged aquatic macrophytes which are mostly used for the phytoremediation process. And from the emergent macrophytes, the mostly used species, aquatic species are Typha latifolia, Scripus species, and the Phragmites and the Polygonum. These are the few species which mostly used for the phytoremediation. Now, the next slide, this slide is showing the potential of various aquatic macrophytes in removal of various heavy metals and various heavy metals from the different type of wastewater. So these are the various species of aquatic macrophytes which are used to treat different types of wastewater and remove different type of metals. And now the conclusion. Phytoremediation using aquatic macrophytes is a promising technology applied for the removal of pollutants from the aquatic ecosystem. These techniques not only eliminate the heavy metals but also removes the organic pollutant like nitrates, phosphates as well from the wastewater. Therefore, it regained the quality of pollutant water before into the environment. This review reveals the numerous features of aquatic macrophytes application in the phytoremediation of various aquatic pollutants. The review also covered the importance of free-floating aquatic macrophytes because of their outstanding hyperaccumulator aptitudes. And the Salvinia and the Pistia species are the two most widely used aquatic macrophytes for the treatment of agriculture, domestic, and industrial wastewater. The large application of this plant is due to their obtainability, viability in a noxious environment, bioaccumulation capacity, invasive mechanism, and biomass abilities. On the basis of the present review, the benefits of using aquatic macrophyte to remove pollutants are vast because this skill does not only eliminate their contaminants but is a low-cost, budget-friendly, aesthetically pleasing, and sustainable to the environment. Now, I would like to acknowledge the author.
Please omit it.
Yes, sir.
Please omit the acknowledgement part. It is already written in your slide.
Okay. So these are the few references which I have used. Thank you.
Okay, Madam Jones, it is,
Thank you, Maria.
Thank you, sir. Thank you, sir.
So next participant, Bhuvaneshwari M.
Yeah, ma'am. I'm here.
My slide is visible, ma'am.
Not yet.
My slide is visible.
No.
Now, ma'am.
Now visible. Make it full slide, huh?
Yes, sir. Good evening, everyone. My name is
Slide
Rohit.
maintain time, please. Huh?
Okay. Yeah, an exclusive review on Serapegia junctia species. I'm going to deal about the species. The plant which have a most phytoconstituents present in this family, the Serapegia junctia only contain more medicinal value. So from this, I'm going to, I reviewed distribution and morphological and phytochemical and pharmacological studies I have reviewed. Then through this 200 species are available here, and according to the IUCN this comes under the threatened category, and as well as the biological source, this plant is belong to Apocynaceae, the different synonym of this plant, and the plant is otherwise known as Somalata. And because why it is Somalata we are calling, because the Soma drink is prepared from this plant, and this is a different vernacular name. Sorry, according to the area it may vary. Then the origin and the geographical distribution, these are the different areas these plants are available throughout the world. Then the habit of this plant is commonly cultivable land, and it is sorry, it is mainly present in fens and forests and hills, and the root is like a tuber, and the leaf is like a lanceolate shape, and the flower is goblet shaped, and it have a purple spots on the stem, and it have a weak stem, and the pollination is limited, and fruit set is under certain conditions. During July to November, the maturation of the flower and fruit is happened, and the fruit is elongated and follicle. Then propagation is vegetative method through the root tubers or stem cuttings. This is the plant of Serapegia junctia. Then the traditional uses of this plant. This plant is mainly used for ulcer purpose. Along with the goat milk, the extract of this plant is given orally to the people, and it is very familiar in the Sirimalai near the Dindigul district. The Balian tribe peoples are giving the peoples as a medicine for ulcer purpose, and it is mainly used for ulcer, inflammation, and anti-cancer, and as well as anti-diabetic purposes. And this is a RET condition of the plant, and we have to protect this plant for future generation. And these are the problem happen because leads to threatened condition. Then what are the phytoconstituents is available in this plant means carbohydrates, saponin, and some kind of phenolic compounds, and fatty acids, flavonoids, coumarin, and the family which contain a familiar and characteristic is serapegin is present, and as well as coumarin is the very common in this plant. Then through the GCMS analysis, the result was given as 29 compound were isolated already, and through the FTER they have isolated these chemical functional groups are isolated. Then the terpenoid, which is mainly used for antiseptic, and these are all the different uses, and polyphenolic compounds of ethanolic extract shown the gastroprotective. This is a gastroprotective and hepatoprotective also, yeah, very familiar to cure from this plant. And tannin also helpful to cure intestinal disorder. Then anthracene glycoside, which is involved in the anti-inflammatory purposes and hypertension, it reduces the pressure, and as well as the saponin and anthocyanin, which cures the healing property, and fatty acids also use different problems. And the in vitro propagation is very well known in this plant because it is in the condition of threatened. So the Serapegia junctia is under TLC purpose. They found the Serapegia in a characteristic phytoconstituent is found from that, and the chemical substance which is used to cure different purposes like hepatoprotective and anti-ulcer. These are the characteristic Serapegin help to cure. Then animal study also revealed the same. Then coumarin also used to cure the different diseases like anti-inflammatory and proliferative and antiviral and carcinogenic. It is anti-cancer activity is found through animal studies. And Serrapegia junctia is found against a different antimicrobial work was done through the different microbes, Pseudomonas aeruginosa and Escherichia coli, then Klebsiella pneumonia. They were used in the methanolic extract. It is they found. Then Klebsiella, they were found against aqueous extract. Then Candida found through that. Then the Serrapegia junctia methanolic extract, it is mainly against the bacterial activities. Then acute toxicity level through this they found up to the maximum level. The animal was safe up to 3000 mg per kg body weight. Up to that it is very safe. After that it is changed into poisonous one. The toxicity of Serrapegia junctia reduces without side effects because it have more medicinal value and it is safe to use. Then anti-ulcer, it is a peculiar study was done. The ulcer was calculated with a different score, and finally they examined it is helpful to cure intestinal, the colon cancer was cured through this plant. So by using the cell line HCT118, the colon cancer was found that. Then anti-diabetic study which reveals that methanolic and ethanolic extract of this plant, both the in vivo and in vitro plants were analyzed, and they found the colon of human, they different action was happened. Then which when the carbohydrate is changed into starch and oligosaccharides also converted, then which contain a different heavy metals like chromium, manganese, copper and zinc, cadmium, mercury, lead, arsenic. These are all the different metals were found through this plant. That was safer by suggested by World Health Organization. So that is used to cure. We can safe to use the plant. And if free radical scavenger activity also done from this one. Then the significant lipid paradoxin, the antioxidant study also found through the enzymatic study. It is useful to cure many diseases. Then finally, this plant is contain many medicinal value as well as which is helpful, which is in the condition of endangered and threatened condition. So we have to protect this plant through different methodology, and specifically through the micro propagation we can protect this, and as well as throughout the world we can propagate and secure this plant for future generation. Thank you all.
Thank you.
Next, Hasanat. Next.
Yes, ma'am. Is my slide visible? Is my slide visible, ma'am, sir?
No.
Oh, wait. Now?
Yes.
Okay. I'm going to present my review work. The review work is based on the topic influence of elevation on the caffeine in tea and coffee. Here is the introduction of tea and coffee. We can get tea from Camellia sinensis, and coffee is another most consumed beverage which we can derive from roasted coffee seeds. Only 2 species are used for coffee. Yes, sir.
Go to the objectives, then material methods and result discussion part. No introduction.
Okay, sir. Okay, okay, sir. The slide is about the objective of this review work. The chemical substance that occur naturally in tea and coffee is caffeine. We drink tea and coffee to prevent drowsiness. So caffeine has so many benefits and disadvantages too. So through this review work, I am going to show whether elevation change affect the caffeine concentration or not. This is the brief concept of caffeine. Um Caffeine is a trimethylxanthine group which is present in tea and coffee naturally. Okay, and yes, this is a table which is about the climatic condition that is
Your
required
slide is
for
not
tea.
moving. Your slide is not moving, so go to the next slide. Next slide, then continue. Huh?
Why slide is not moving?
Slide is not visible.
Slide is visible, but in first slide.
Escape.
Yeah, that
Now?
is going on. Yeah.
Okay. Okay, sir. So this is the brief concept on caffeine. Caffeine is a purine alkaloid and methylxanthine group which is present in tea and coffee. This table is about the climatic condition. Sorry. This table is about the climatic condition that is required for tea cultivation in different countries. In India, cultivation is occurring at the vast range of elevation. The range is starting at 60 meter to 2623 meter, which is a noticeable thing. The highest range of elevation for tea cultivation
Slide
is
is
in
not
Kenya.
moving. Please move your slide.
Why, ma'am?
Yes, go.
Okay, the highest range of elevation for tea cultivation is in Kenya. As we see the requirement of temperature in Kenya, it is 16.4 to 18.1 degree centigrade, which is lowest, and in other country, the temperature is moderate. There is huge difference in annual rainfall in Sri Lanka. Here tea cultivation is occurring 2 to 3 to 4 17 meter, which is higher than other country. And the next slide is about the climatic condition that is required for coffee cultivation in different countries. Here we can see in India, coffee is produced at higher altitude, and the lowest temperature is seen in Brazil and the highest is in Vietnam. The annual rainfall is moderate in all the countries. And then this is some benefits of caffeine. Caffeine stimulates the central nervous system. Caffeine can prevent our drowsiness, and caffeine has so many disadvantages too. More taking of caffeine can cause dizziness, insomnia, and this now coming to the discussion part. The table is about the caffeine content in tea from different altitude. In India, at Darjeeling, at the higher altitude, the caffeine concentration is higher, and lower elevation, the caffeine concentration is lower. So, the same thing is going for China and Sri Lanka. Another thing is, in Sri Lanka, the overall caffeine concentration is higher than the other countries. This may because of the climatic condition, as we have seen before that in Sri Lanka the annual rainfall is much higher than other country. And this table is about the caffeine content in coffee in different elevation. Here we can see at the higher altitude the caffeine content is lower, and the lower altitude the caffeine concentration is higher. And now coming to the conclusion, The altitude at which a tea or coffee plant is growing influences almost every aspect of its development from its aroma to nutritional value. So, change in elevation can drastically change the environmental condition, thus affecting the quality of tea and coffee quality. The variation are applicable even in a single growing region. In different altitude, the temperature is also different. Temperature and precipitation is also a very important factor for the tea and coffee production. uh, so when the altitude increases, the air becomes colder, soil becomes rockier, and due to the land gradient, rainwater is less available here. This long and slow growing process is very suitable for high quality of tea. So, besides the height of region, range such as temperature and rainfall both affect the soil fertility. And uh, so, if we omit all the exceptional if we omit all the exceptional cases, we can conclude that whichever the country is, the caffeine level is always increasing with the increase in altitude, whereas opposite scenario is seen in case of coffee. That is, caffeine level is decreasing with the increasing in altitude. So as coffee has some disadvantages too, so nowadays is we are so much concerned about caffeine free beverage. If we know which elevation has less caffeine, then in that elevation we can cultivate tea and coffee. So here are some references which I have used, and lastly, thank you.
Next presenter is Sajita Cyril. Sajita.
Okay. Good evening, everyone. I'm Sajida Cyril. I'm doing my master's in forestry from Uttar Bengal Krishi Vishwavidyalaya. Here, I'm going to discuss about our paper, Reviving Urban Forestry for Sustainable Cityscapes, an insight to the refuge of existing natural resources. More than half of the world population shares space and resources in urban areas. With the overflowing population and change in land use, depletion of natural resources are happening in cities. According to the UNEP report 2021, it shows that cities are heating up at twice the global average rate. And according to the World Air Quality Report, the PM concentration, 2.5 concentration, and air pollution are high in cities, and New Delhi leads the list of capital city ranking in the air pollution. So in this scenario, urban forestry emerges as a long-term strategy to protect the natural resources from different threats in the urbanized locations. So urban forests are woodlands and other tree components in the urban areas which are dedicated to enhance the sociological, physiological, and economic well-being of urban society. These are the key roles of urban forests, which help in carbon sequestration, biodiversity maintenance, microclimate amelioration, temperature maintenance, runoff reduction, and air pollution and noise pollution abatement. In addition, out of the 17 Sustainable Development Goals, urban forests contribute about 9. The current study reviews the role of urban forestry in the sustainable development of cities with a special emphasis on the natural resources, soil, water, air, vegetation, and wildlife. So we followed rapid literature review method, where literatures are searched using keywords urban forestry, urban greening, natural resources, and sustainability from the databases Google Scholar, Science Direct, and ResearchGate. About 155 papers are collected. From that, duplicates are removed, and after title and abstract screening, 52 papers were undergone full text read, and data are extracted and meta-analysis were done using Microsoft Office packages. So the meta-analysis showed astonishing results. The figure which shows that the trends in publication frequency on the urban forestry related articles. So it was found that the research frequency has increased sharply over the last few years, attributed to the concerns of the global climate crisis and enhanced interest in the sustainability and increasing popularity of the urban forestry. And we also categorized all the papers based on the discussed aspect of the urban forestry. Most of the papers discusses the benefit and ecosystem services of the urban forestry concerning the natural resource conservation. Air and water are studied more compared to the soil because they directly impact the quality of the urban life. Another interesting factor we found is that the latest paper regarding urban forestry interpret the qualitative benefits through quantitative data analysis, which indicate the advancement in research and technology. These are the ecosystem services provided by urban forest. So it provides all the types of services, which include provisional, cultural, regulating, and supporting services. So regarding their contribution to their natural resource conservation, urban forest regulates the air quality by mitigating the air pollution by lowering the gaseous and particulate pollutant concentration. It also helps in carbon sequestration and reduces the carbon dioxide concentration, which is a major greenhouse gas. And it also helps to control air temperature and noise pollution. Concerning the water resource, it regulates the city's hydrology. With the deep root system, it will increase the infiltration rate and increase the groundwater recharge. And it also acts as a biofilters and purifies the water. And the tree components in urban forestry provide variety of habitats, shelter and food to organisms, thus enhance the biodiversity of the cities. And also by preventing the soil erosion, it conserves the topsoil in cities. As a conclusion, the urban forests are suitable in enhancing the sustainability of cities. Even though the concept of urban forestry is relevant in this modern context, it is not widely popular and still ignored in urban planning activities. So new policy interventions and awareness must go in tandem to extend the research outcomes to benefit the immediate community. These are the references. Thank you so much.
Thank you, Sajida. Sir, any question or any discussion?
No,
Sir.
I think question can be raised in the platform, not in the platform, but they can ask in the through email because time is very.
Yes, sir. Again, some questions are here in the chat box. Participants are asking to not leave the meeting. Try to answer this through chat box, if possible. So next presenter,
Yes,
Gautam Ghosh.
that is good.
The next presenter, Gautam Ghosh. Are you ready?
Yeah, ma'am, I'm ready.
Okay.
Respected sir, today's my topic of presentation, role of plant bioresources in traditional healthcare system, a case study from Aparsiam district of Arunachal Pradesh. Bioresources include all products and services coming from the natural environment that satisfy the needs and wants of humans. The survival of mankind depends upon the availability of bioresources and their proper management. It is also a major component of the natural resource that occupy a central position in the tribal culture and economy. Ethnic groups across the globe possess tremendous amount of traditional knowledge, most of which are poorly documented and hence are largely unknown. This knowledge was passed on through generation and it played an important role in the conservation and sustainable use of biodiversity. Some notable progress has been made in the field of, now I want to say some plant bioresources and ethno-medicinal practice is the belief and practice relating to health and disease that are products of indigenous cultural knowledge and of the particular communities. Studies have been carried out globally to verify their efficacy, and some of the findings have led to the production of plant-based medicine. Some notable progress have been made in the field of ethnomedical research in the tribes of Arunachal Pradesh. However, little information is available in connection with composition of ethnomedical plants by research in Upper Siang district of Arunachal Pradesh. In this paper, an attempt has been made to deal with the composition of ethnomedicinal plant diversity with indigenous medicine for some common diseases practiced by the indigenous people of this Upper Siang district of Arunachal Pradesh. The present study extensive survey was conducted in 15 villages of Upper Siang district of Arunachal Pradesh. Among the 150 household, and that is dominated by Adi Idu and some Buddhist tribe like Memba Khamba. The village headman and herbal practitioner were interviewed and various uses of plant as well as their vernacular names and other relevant data collected. During data collection, the informal discussion was held in the village headman, medicine man. Finally, the ethnobotanical market survey was done to assess the commercial viability of certain species, and information about particular use also collected. Now, observation. During my collection, I observed some plant and animal component. Those are contributing for healthcare of this particular area, and this table shows the plant composition in that particular area, whereas some animals also contribute for healthcare system, as one of them is Mithun, sloth bear, Mark's deer, Himalayan black bear. This animal also use with the plant resources for curing different kind of diseases or ailment. Now, sharing of bioresources in healthcare system in the study area, it is found that 89% plant and only 11% animal contributed the healthcare to the healthcare system of that particular people of that region. Plant-wise contribution to cure disease in the study area, it is shows that highest the plant Xanthogylum acanthopodium shows that the highest number itself can cure highest number of disease, that is 6, and in some cases single plant can use for treatment of a specific disease. If we consider about the plant part used for treatment of disease, then we see fruit is the major component of the plant, followed by leaves, and likewise tubers, bulbs is the least. In the preparation of different kind of medicine, traditional medicine which is used by the local people. Now, species richness, family-wise species richness of that particular area, it is found from our study that a single family maximum species, that is 2, contain 2 species, can contribute highest number of plant component for preparation of medicine. Now, species composition of traditional medicine. There are 33 plants identified which is commonly used for the treatment of disease, but it is also subgrouped in different categories, like some species responsible for curing blood pressure, some species responsible for curing boils, jaundice, some species. In this way, we got 8 species responsible for diarrhea, dysentery, and stomach, and this is the common problem in that area, and followed by 6 species cut and nodes. It is also common problem to that particular area. Likewise, they also use some species for snake bite, losing of high fat problem, overcome of high fat problem, fever, etc. Now, the composition. Now here we see cart and oats. This is a common problem in that particular area. The forest dweller infected by this problem severely and many times. So they also use different type of plants from their forest only. As for example, Rixinus communis, where latex is used in the cart and oat area. Likewise, Azaratum conigioides. In this way, Hedychium spicatum, Bujolgia, and in some cases, large species of bamboo species is also used for healing or check for blood clotting of blood. The epidermal part of the pseudostem is used for this purpose. Diarrhea is another important problem. Here also, large number of plant species also be used to treat this problem. Begonia palmata, Gracinia pedunculata, Calicarpa arborea, Gingiva officinale, Solanum torvum, Rixinus, and the species of Smuza in various way, either alone or in combination with other plant species among these plants. Likewise, fever.
Sir, try to sum up, sir.
Okay, ma'am. There are so many. Okay, just a minute. Single species also shows multiple application in healthcare system. As for example, Xanthogella macanthopodium, another species, Mismithita, that is Coptis in this way, plant are used there here. Now, conservation and conclusion. A large number of plants used in treatment of different type of ailments for malaria, cart, wound, swelling, skin disease, gastrointestinal problem, antidote of worm, etc. And inventorization of such bioresources will go a long way in giving prosperity to mankind. Medicinal plant used in ethnic community could be considered as a substitute of allopathic medicine. Coptis, Xanthogella macanthopodium are the common medicinal plant used by the tribes. Among these, some species cultivated and maintained by the people include the famous Mismithita, that is Coptis by the Idus. They cultivate the Theta under forest canopy. In this plot, they do not cut the trees, but only clear the ground flora to facilitate the plant growth. Further, the huge genetic diversity in various ethnobotanical plant species have vast scope for collection and conservation in situ and ex situ, both in the state. Thank you.
And thank you, Gautam sir. And your questions are in chat box.
Okay, ma'am.
Okay, sir. Due to time constraint. Okay. So next presenter, Dr. Ravindra De.
Yes.
Ravindra De. Ravindra De, sir.
Yes. I am presenting.
Bhattacharya sir, I am really sorry.
It's okay.
I have to announce because of time constraint. Please try to show your findings and result parts only.
Respected chairperson and other dignitaries, I am presenting on topic spice as natural products and their medicinal values. Spice is a dry seed, fruit, roots, barks or vegetable material used in nutritionally in
Slide
amount
is not
as a food
visible.
supplement. Some spices are reputed to possess several medicinal and properties and hence find position in the preparation of number of medicine.
Ravindra, your two devices are nearby, so keep the devices at least 1 meter gap. That's why actually noise is coming. Both are actually open. At least mute on.
Okay, sir. Bay leaf is one of the production fresh bay leaf. Phenol was the major component together with alpha-amino acid. So some problem here.
Sir, please don't waste time.
Your voice is not coming. So continue, Ravindra.
Ravindra De, sir. Are you in the meeting, sir?
Dr. J. So if slide is not Dr. J.
He's on mute. He has muted.
Dr. J, please carry on. Or if slide is not coming, you can brief by orally. You can brief it by orally.
Ravindra De, sir, please conclude. Your voice is not coming.
Sir, please mute yourself, unmute yourself. Dr. Dev.
Then we should move to next presenter.
Yes.
Next, Ankita Datto. Sir, Ravindra De, sir, stop presenting. Next presenter, Ankita Datto. Ankita, are you here?
Problem here, sir. Okay.
Ankita, are you here?
Yes, ma'am. Am I audible, ma'am?
Yes.
Okay,
Yes, you are.
I'm presenting. Am I visible, ma'am?
Yes, it is visible.
Yes. Okay. Very good evening to the respectable dignitaries and the organizer and the elite listeners. I am Kita Datta, scholar of Dr. Nandini Ghosh, Vidyasagar University, Department of Microbiology. Want to throw some light on the topic, study of the pollen diversity of the vegetation of Midnapur town during the post-monsoon season. So we know that pollen grains play a very vital role for the gene flow, and studying of the pollen grain is very important for assessing the biodiversity, the plant-pollinator interaction, and also to understand the impact of climate and pollution on the plant population. And we know that pollen grains are known as seasonal air pollutants, which can trigger allergies. And people suffer from allergies during certain period of time, often gets exaggerated during the pollen showering period. And we know that Midnapur town, the district town of Paschim Medinipur, is covered by varied kinds of vegetation, starting from herbaceous ground cover to shrubs to trees. And not only that, during the post-monsoon season, people often suffers from different kinds of viral infections and different kinds of flu. So it is quite unnatural that not only there's flu, but during this post-monsoon season marks the onset of pollinosis of different kinds or wide variety of plants, which can also contribute to the allergenic effect of certain atopic population. So my aim of the study was to conduct or assess the vegetation of Midnapur town, along with providing the records of certain pollen allergens and analyze them through light microscopy and scanning electron microscopy. So as I said, Midnapur town was selected for the study because of its wide vegetation cover. The exotic species of herbs, shrubs, and trees were noted, and vegetation analysis was done, followed by the collection of the pollen grains, and was subjected for the light microscopic and the scanning electron microscopic study. For light microscopic study, the plants were the inflorescence from the flowering plants, those who actually flowered during the time of October and November. Extensively, the inflorescence were collected, followed by the isolation of the anther. It was kept in the freezer, and then for the light microscopic study, these anthers were crushed in acetic acid, centrifuged and washed, followed by dissolving them in 10% KOH solution, and was kept in water bath for 80 degrees centigrade until the solution turns a bit brownish, and followed by centrifugation. Then it was washed thoroughly, bleached with 10% HCl solution, and then the pollen grains were mounted and was mounted in glycerin and was viewed under the light microscope. And for certain pollen grains where the exine ornamentation or the external features were not visible, it was subjected to scanning electron microscopic study, where the anthers were dried, sieved, and the pollen grains were isolated. It was brushed onto the platform, and it was prepared for the sense studies. So, as I said, that the vegetation of Midnapur provides a wide variety of plant species contributing to the biodiversity. And during this month of October and November, this land gets covered with many herbaceous species contributing to the plant population, out of which the plants that I found during the flowering time of the October and November was Lindania crustacea. Then there was Sida, then Eragrostis tenella, Polypogon, weed like Triumphetta, Mitracarpus hirtus, Cynodon dactylon, Scoparia dulcis were noted in the flowering condition. Not only this herbaceous ground cover, but also hard shrubs were also noted, out of which there was wide variety of Acacia sophera. Wide variety of acacia was found, out of which Acacia sophera, Acacia tora, there were Acacia occidental was noted. Not only that, Crotallaria pallida, then other species of Crotallaria was also found. Beside this, there was the vegetation which was noted comprised of Hibiscus vitifolius, Urina lobata, Mimosa pudica, and Acacia sophera, and they had a good flowering period during the time of October and November. So not only upright trees, but also lianas were also noted down, where Mycenia micrantha formed a part of the vegetation cover, with Lupa cylindrica and Clitoria tarneti was also noted. And the hardy wood trees were also found, out of which mentioned here is acacia, Madhuka indica, Azadirachta indica. Few of them were not flowering during that time, but it was spotted and was noted, like Ashoka, Terminalia, etc. Soria robusta was also there. And out of all the kinds of plants that were noted, a few of the reported allergenic plant species were also found, out of which there was Eucalyptus globulus, because it forms almost 30% of the land cover of the Midnapur town, forming the dry deciduous type of plant population. Then there was Piltophorum pterocarpum and Delonix regia. These are already reported pollen allergens by many researchers and many researchers. So this was this is a picture that was being taken after the isolation and the light microscopic study, where the pollen grains were clearly visible, and the pore condition or the external structure, the shape of the pollen grains were completely visible. And we found, out of which I can say that outer external ornamentation was also clearly visible in case of Nympha cylindrica, Tridux procumbens, where the tectum ornamentation was pretty clear. Not for which plant species where the external morphologies were not quite visible, or the pictures that was not quite good enough to understand what is the outer ornamentation, same studies were performed, out of which we can see that the Cassiophyra, the pollen grains of Coccus nucifera, Rostoria robusta, and Lantana camara, the outer ornamentation or the tectum ornamentation was plain. There was no conic ornamentation. The surface was plain, which we can say as select type of tectum ornamentation. In case of hibiscus pollen, we found that there was spike-like structure that developed on the tectum, that is the tectum ornamentation, which we can call as pinnulate. In case of Amaranthus, we got perforated tectum layer, which is actually termed as punctate. And in case of the most allergenic pollen grains that were allergenic pollen grains, or the, I should say, that allergenic plant species that are already defined, that Peltophorum pterocarpum and Delonix, yes ma'am, Delonix regia, there was tectum ornamentation was reticulate. In case of grass family, we found that the outer layer was sunken. So this is these are the nature of pores or the colpi that I found out after studying the light microscopy through light microscopy and scanning electron microscopy. I'm just not going into it. So what can I discuss from the above work that I have done? That villagers gets the villages of the town of Midnapur district gets quite availability or easy access to the forest, cutting down the trees, hardy shrubs for the firewood purposes, and clearing the forest cover from turning them from dry deciduous type of forest vegetation to a xerophytic condition. And not only that, when reported plants which are regarded as potential allergens or allergen causing agents were also found, and this post-monsoon season marks the onset of various kinds of flu and fever, which is quite misleaded by other kinds of allergenic symptoms that is experienced by the allergenic population or atopic person, having hay fever, runny nose, watery eyes, and respiratory troubles. So all these symptoms are still unknown by many people. People have less knowledge about this. So my future plan for the study is to undergo the biological study of the pollen grains that is present in the air, and to find out the allergenicity of the pollen grains present in the air, which is already reported or can be regarded as a potential pollen allergens. So I conclude my topic by saying that the study of the pollen grains from the local vegetation and their pollination and their pollinating season is very important and gives us information about the plant taxonomy, the evolution, and as well as not only this, but also it gives us prior knowledge about the pollen grains or the bioparticle present in the air, which can cause allergy. And this is the preliminary step of identification of the pollen grains, forming the plant population of that particular geographical area. And due to the marked increase of cough and cold, the related symptoms during this post-monsoon season can also be a correlated factor with viral infection and allergic symptoms. So this study provides a basic information about the vegetation and the plant cover of the post-monsoon season. Further study will open up new avenues in the research and can give us prior idea about the allergies among the local population. These are the references that I took. Thank you.
Thank you, Ankita. Next presenter, Deepu Samanto.
Am I audible?
Yes.
Before,
Yes,
within a
sir.
limited time, continue, just objectives, methodology, and result.
Okay, sir. Is this visible?
Yes.
Okay. Good evening, one and all present here. Myself, Dr. Deepu Samanto from Department of Botany, Dr. Kanilal Bhattacharya College. The topic of my today's presentation is plants for soil pollution control, a review. Soil, the upper layer of the earth, is the main and natural medium for plants' growth. It is mainly composed of minerals, organic matter, soil water, and soil air. Soil pollution is a major problem for us because polluted soil has different adverse effects on environment as well as health. Soil pollution is harmful to living organisms as well as it decreases the activity of soil microorganisms also. There are certain causes of soil pollution like chemicals, radioactive materials, toxic gases, heavy metals, and chemical fertilizers or pesticides also. All these causes of soil pollution has the adverse effects like pollution of underground water. These are the toxic to plants and having the adverse effects on plant growth and metabolism also. There are certain harmful effects of soil pollution to health of human beings, birds, plants, and certain microbes also. Now, as there are so many adverse effects of soil pollution, so we have to move in the several methodologies to protect the soil pollution. Some of them are crop rotation, strip cropping, and we can use certain plant-based grain manure instead of chemical fertilizers like Azadirachta indica, Calotropis, etc. This type of organic manure increases soil fertility and also improves the soil structure, and thus this control the soil erosion also. After that, there is important role of afforestation. Afforestation plays a major role not only in controlling the soil pollution, but it affects the it has the role for controlling all types of environmental pollution. Afforestation helps the maintaining of pH of soil. Planting trees also helps to prevent soil erosion and it enhances the soil fertility. It is a sustainable soil management technique. Now, up to this, it was a basic aspect for soil pollution and kind of prevention. Now, in this review, I am mainly focusing on the plant-based approaches to control the soil pollution, that is phytoremediation. The term phyto means plants and the remediation means to cure or restore. Here, the plants can remove pollutants from soil. The root system of plants play a significant roles in controlling the pollution by accumulating the certain heavy metals, and there are certain types of phytoremediations like phytoextraction, phytostabilization, phytovolatilization, phytodegradation, and rhizodegradation. This is the diagrammatic or pictorial representation of different types of phytoremediation that we can use to remove the soil pollution. Now, first, phytoextraction. More than 400 to 500 plant species are reported, mainly from Brassicaceae, Fabaceae, Poaceae, Cyperaceae, and from different families also. As per example, Nicotiana tabacum can accumulate heavy metals like cadmium in high volume in their leaves. There are certain examples also like Ipomia, rumex, etc. Now coming to the phytostabilization. In this method, the metal tolerant plants are used to reduce the bioavailability of heavy metals in the soil. Examples of the plants are Typha, Brassica, and Euphorbia, etc. Now, another type of phytoremediation is phytovolatilization. Here, the plants are used to take up heavy metals from the soil and release it into the atmosphere in a volatile form. Certain plants are used for these purposes, like Medicago sativa, Phragmites, Pteris, etc. Plants under Brassicaceae family, such as Brassica, has the ability to take the toxic pollutants from soil, and they can convert them into a less toxic volatile forms. Now coming to another way, that is phytodegradation. Here plants can degrade organic pollutants in the soil. Now, another type of phytoremediation is the rhizodegradation. In this type of phytoremediation, the rhizosphere region plays a vital role to degrade the contaminants. Plants like Casanus casana can use for these purposes. This type of plant can degrade the rhizospheric the different contaminants from the rhizospheric region. Now, this is the summary table where I have plotted so many plants and the methods of each plant by which they can control the soil pollution. Now, from this review, I can conclude that use of plants is the most effective way to control the soil pollution. There are several methods to mitigate the soil pollution, but using plants for soil pollution to control is the best and sustainable way. We need to remember that pollutant-free soil gives the healthy food and ensure the food security. It is the responsibility of all of us to keep the soil pollution free in order to build a safer world for future generations. Now, these are some of the references that I have followed to prepare this review. And now I would like to thank the organizing team, my college authority, and all my teachers. And special thanks goes to the sir, that is Dr. Devabrata sir, for constant encouragement. Thank you all.
Now, next presenter is Dr. Mitu De.
Is the slide visible?
Yes.
Okay. Chairperson, sir, dignitaries, and other presenters, a very good evening to all of you. The title of my talk is Variation in the Grain Morphology Among the Dwindling Indigenous Rice Biodiversities. It is with my co-author, Dr. Shanti Ranjan.
Excuse me, madam, your slide is not visible.
The slide is visible.
Is it visible now?
Slide is visible, but you just keep safe distance between two, because two devices are open. So
yes. Okay, so I'll just very quickly say there's something wrong with my microphone. Okay, so after the Green Revolution, there's a lot of genetic erosion of the indigenous rice varieties. And this is the definition of the rice land races, and I have taken 15 rice land races for these studies. These are the names, and I was just looking at the different grain size. So the material methods that was followed by the standard evaluation system of the International Rice Research Institute of Philippines. And these are the different descriptors that were used. And many of them were about because the apiculus color, the awn, the seed coat color, these were taken. So these are some of the findings that happened. So you can see from this picture that all the grains are different. And these are some of the findings. And this is just the terminology, which is the lemma, paleo, and all. So the variation that I got, because it was a descriptive code, it is marked in the codes 0, 1, 2, 4, and so these are some of the variations noticed. The color of the grain is different in most of the varieties. So usually it's yellow, but some of them you're getting red, black, and there's a variation in the kernel shape and color also. So different colored kernels, and so this is all there. The variation in the shape and size of the kernel and the grain. So this is a picture of the different. And the discussion is that there's a variation in most of the Anderson studies, and so the conclusion is the genetic erosion of the steady loss of genetic diversity of the on-farm agriculture is actually the key threshold of sustainable management. So we need to document these varieties before they are become extinct. So I'm very sorry for the microphone and its there. So I come to the end of my presentation. Thank you.
Now, Shirley CS. Your turn is now. Shirley CS. Shirley?
Ma'am, yes, ma'am. Yes, ma'am.
Okay.
Ma'am, is my slide visible, ma'am? Ma'am, is my slide visible, ma'am?
No.
Dear. Is now my slide is visible, ma'am?
Yes, Shirley.
Okay, thank you, ma'am. Good greetings to everyone. Myself, Shirley, PhD scholar from Department of Botany. My research topic is pharmacognostic phytochemical analysis and free radical scavenging activity of Callistemon citrinus leaves. And moving on to introduction, plants are the reservoirs for the possible and useful compounds and applicable for the needs and drugs were resistant to some diseases. There is a need to come across and advance brand new phytochemicals in plants that can be used for several therapeutic uses. So medicinal plants and herbs are of great significance to the human health all over throughout the world, which consider an imperative source of powerful therapeutic agents to treat an extensive range of ailments and health disorders. So the present study has been undertaken to evaluate several characters and parameters of Callistemon citrinus leaves in order that the authentic plant material might be explored for its therapeutic uses. Callistemon citrinus belongs to the Myrtaceae family. However, cosmopolitan in Asian countries, in Tamil Nadu, it is referred to as Pallosa tree as a result of its spiky inflorescence that resembles a bottle brush. So the aim of the study is to standardize the pharmacognostic evaluation of Callistemon citrinus, to analyze the qualitative and quantitative phytochemicals, to analyze the antioxidant activity of the selected plant material. So these were the different methodologies which was utilized for the research work. Pharmacognostic study, phytochemical study, antioxidant activity analysis. And moving on to the findings of the study, coarse powder of the dried leaves was used to study the organoleptic characters, which includes the macroscopic appearance of the drugs, such as color, odor, taste, sometimes feel of the drug to touch. So based on the characters, the results were tabulated, and the physicochemical properties of Callistemon citrinus, such as loss of dry foreign matter, total ash, acid soluble, water soluble moisture content, were determined as per official methods of the WHO guidelines on the quality control methods of medicinal plant materials. Moving on to the qualitative phytochemical analysis, the curative properties of medicinal plants are mainly due to the presence of various complex chemical substances of different compositions which occur as secondary metabolites. So the leaf extracts of different solvents were screened for the phytochemical compounds using standard methods, and the details of the investigations were mentioned. So the Callistemon citrinus leaf extracts was evaluated at different concentrations like 10, 20, 40, 60, 80 and 100 mg per ml for their capacity to scavenge DPPH free radical along with ascorbic acid as a positive control, methanolic leaf extracts of calcium citrinous leaves observed more antioxidant activity when compared to the ascorbic acid. So, conclusion, the findings of the present study suggest that natural products are yet reflected as a potential source for the drug discovery and therefore play a vital role in drug development programs. So, the leaves of Callistemon citrinus selected for the phytochemical, physicochemical, and biochemical investigations. The outright findings of this study serves contribution in proper use of these leaves for superior healthcare system of people and provide an insight into the usage of Callistemon citrinus leaves as a good source of naturally occurring antioxidant, and it is suggested as the part of phytopharmaceutical importance. So, thank you for the opportunity.
Thank you, Shirley. Then N. Lavanya. N. Lavanya.
Yes, ma'am. Yes, ma'am. Good evening, ma'am. Good evening to all present here. Is this my slide is visible?
Yes.
Yes.
Okay,
Okay,
yes.
ma'am. Thank you. Myself, Lavanya, research scholar from Botany Department. I'm happy to present my project work entitled A Survey on Floral Biodiversity in Avinashlingham Institute for Home Science and Higher Education for Women, Campus 1, Coimbatore, under the supervision of Dr. Leena Lavanya, Assistant Professor, Department of Botany. A survey in order to update data and information on biodiversity in Avinashlingham Institute Campus 1. Various forms of taxonomic literature have been incorporated for proper identification of unknown plants. Some of these are among the oldest and most complicated literature of science. Here, some of these are, I have shown here. Theophylatus, who is the father of botany, has identified the known plants in his name of De Historia Plantarium. Pedanicus Decorlius identified and described the medicinal plants in the name of De Materia and Medica. Carolus Naes, who is called as a father of taxonomy, classified the plants in the name of species Plantarum. William Roebuck, considered as a father of Indian botany, published the plants in all Coromandel Coast. So, and this most of the eminent person have published the biodiversity of floral. Here I shows the objective of my study. Survey and collection of plants from the study area, identify the species collected from the study area, describe the identified species, preparation of the detailed account of a plant found in the study area based on own collection, consultation of the herbarium and study of relevant literature. Preparation of the comprehensive list of economically as well as medicinally important species for their conservation and sustainable utilization. This slide shows the geographical location of my study area. Flora means inventory of plants of a definite geographical region. It is the important forms of taxonomic literature on which study and research on plant taxonomy are highly based. These are not only essential for proper identification, but also helpful to understand and conserve the biodiversity. The most important part before indicating work for a flora is to select the appropriate area of study which determine the scope and value of flora. So here I have chosen Coimbatore city, which was located in western Tamil Nadu state, southern east of India. It is also known as Manchester of South India. This slide shows the study area of our study. I have chosen Avinashlingham Institute of Home Science and Higher Education for Women. It has two campus, in which I have chosen campus 1 is situated in the heart of the city. It was established by great patriot and educationist Dr. T.S. Avinashlingham in the year 1957. It has various buildings and it consists of palms, climbers, herbs, shrubs, trees, which give shelter and food for many faunas, along with clean air and shades. A survey was done in campus 1 to find the number of angiosperm and gymnosperm. This area The area which I have chosen were rich in floral biodiversity. This slide shows the methodology of my study. Ground survey was done extensively from December 2007 to March 2018 in Avinash Lingam Institute of Home Science and Higher Education for Women, Campus 1. The plan shows the plant species were identified with the help of keys provided in the floor of Presidents of Madras, Campbell and Fisher in the year 1935 and literature available in our library. Complication of data has been analyzed by field survey, collection, study of available literature, field observation during survey, include noting down vernacular name and other collection data, taking photographs, preparation of description, illustration and identification of collected specimen. This slide shows the result of my study. As a total of 1,233 plants were recorded in the entire campus 1, which include many trees, shrubs, and climbers, among which 84 taxa were identified. A total of 73 species representing 65 genera belonging to 78 families have been recorded in our present study. Among this 84 plant, 81 belonging to angiosperm. In that, 5% were herbs, 17% were shrubs, 77% were trees, and 0.3% were climb. While 3 were recorded as a gymnosperm, 79 were recorded as an dicotyledon, and 5 were in monocotyledon. This slide shows the list of plants that were available in Campus 1. In this, most of the families, approximately 61, were represented by single species, that is monospecific family. Among the survey list, Fabaceae floral biodiversity was represented by the highest number of species, that is 8 species, followed by Apocynaceae, that is 7 species, and then Caesalpinaceae, 5 species, after which Araceae, Bignonaceae, Rutaceae, each of which contain 4 species. Then Anonaceae, Asparagaceae, Laminaceae, Malvaceae, Myrtaceae, each of this family contain 3 species. After which Acanthaceae, Euphorbiaceae, Meliaceae, Nyctanthaceae, Rubiaceae, and Sapotaceae. Each of this family contain 2 species, and the remaining 21 families were represented by a single species. As I concluded that various floral species that grown in the area of Campus 1 also have values and direct benefit for the community, both for those who are residing inside and around the area to support our daily lives. The utilization of this floral species are grounded according to their uses. Hopefully, this report may be useful, especially for the strengthening of the management of Avinash Lingam Institute of Home Science and Higher Education for Women in Campus 1 Biodiversity. Thank you.
Thank you.
Yes, ma'am.
Lavanya.
Yes,
and
ma'am.
then the next presenter is S. Manjula Devi.
Hello.
Hello. Yes, Manjula.
Present your screen. Yes, ma'am. Yes, ma'am. Start counting.
Just send your slides, ma'am.
Yes, ma'am.
Sir, Das sir,
Yes, madam.
can we do the announcement in the meantime?
Yes, sir. Yes, madam. Yes, madam. Yes, madam.
Okay. Okay, dear participants. Sorry for the late evening discussions. So we would like to wind up the session with the last presenter, Manjula Devi. And we would continue the technical session 3 tomorrow sharply at 6:30 p.m. So we would like all the participants and all the presenters to join us tomorrow at 6:30 p.m. so that you can complete all the sessions and fill up the feedback link also to get your certificates. Sorry for the inconvenience. Thank you.
Share
Debabrata Das sir, can you hear me?
Yes, yes.
Sir, actually tomorrow I have some issues, so can I present now? I want to take much time, only 2 to 3 minutes.
I want to take much time, only 2 to 3 minutes.
Is this
Today is not
possible, sir?
possible. Gayatri ma'am can tell. Gayatri ma'am.
Ma'am, Gayatri ma'am.
Yes, sir. Yes, sir. Yes, sir.
Hello,
Ma'am,
ma'am.
sorry, ma'am.
Shall I
Ma'am,
send?
just a minute, ma'am.
Yes, ma'am.
You send it. You send it
Shall
to
I?
my mail. I'll share.
Okay, okay, ma'am.
Yes.
Okay, ma'am.
Ma'am, sorry, ma'am. Tomorrow, today we need to wind up with the reporting as well as the vote of thanks for the today's session. So we would like to help you by giving you the first session tomorrow by 6:30. We can present it.
Manjula Devi.
Yes, ma'am.
Yes, ma'am. I'll share now.
Ma'am, I'll share now.
Ma'am, just check and tell, ma'am. Hello, ma'am.
I will present, ma'am. I have received. I will present now.
I will present, ma'am. I have received. I will present.
Okay, ma'am. Okay.
Yes, you can do.
Good evening to all.
Yes, you can do.
Okay, ma'am. Myself, Manjula Devi. I'm going to tell about green synthesis and characterization of silver nanoparticles from Lycoperdicum esculentum, variety PKM1. The nanotechnology is an important field of modern research dealing with synthesis, strategy, and manipulation of particle structure range from approximately 1 to 100 nanometer. Recently, nanoparticle synthesis is among the most interesting scientific field. Silver nanoparticles have greatly drawn the interest of many researchers because of the unique physical and chemical properties that makes them find use in various applications. It has been reported that silver nanoparticles are active against HIV-1 and inhibit HIV-1 replications. Objectives of my study is to synthesize silver nanoparticles from Lycoperdicum esculentum and characterize them using scanning electron microscope. Now, going to methodology. 1 ml of plant extract was added in 20 ml of 1 mm aqueous solution of silver nitrate. It was heated for a few seconds. Later, the reduction of silver ions was observed by color change from yellow to brown. The characterization of silver nanoparticles was carried out by SEM and EDAX analysis. This slide shows synthesis of silver nanoparticles solution. The same image showed the high density nanoparticles synthesized by tomato plant extract and were relatively spherical in shape. The first image of same image of the sample of control tomato plant confirms the extents of very small and unique spherical. The size range between 13.16 nanometer to 33.49 nanometer. The second image of same image of tomato plant treated with mixture of organic and bio fertilizers showed the nanoparticles range from 14.03 nanometer to 64.22 nanometer. This slide shows the peak of element present in Lycopodium esculentum of control plant, and second one is element present in Lycopodium esculentum treated with mixture of organic and biofertilizers. The ADAX analysis showed the presence of various elements. The elements present in the control tomato plant were carbon, oxygen, silver, chlorine and gold. Apart from these elements, the mixture of organic fertilizers and biofertilizers treatments showed the presence of silicon at 2% level. The percentage of silver in tomato plants treated with mixture of organic and biofertilizers was found to be higher when compared to control plant. The presence of silver showed the plant could be used for the production of particles at nanoscale that has a wide range of application in industry. In the present study, green synthesis of nanoparticles from tomato plant is just a preliminary work carried out to analyze the efficacy of the plant in synthesizing nanoparticle. This could be of immense use in medical field. Further studies are required to prove the use of nanoparticles in the medical field for drug development. Thank you. Thank you, Anandal.
Thank you, Manjula Devi. So, thanks to all budding researchers and experienced researchers also for your enthusiastic participation and presentation. I am really thankful to our chairperson and experts for giving us so much time after late night today. So, thanks to all. And one thing I have to say today, that yesterday one reporting was wrong. Actually, Ramastri came was a teacher, assistant professor. She was not a student. Actually, it was mentioned, and she wrote to me that it is wrong. So sorry to Ramastri. If you are here, then we are sorry really. Then it is next to the Gayatri madam.
Yes, ma'am. Yes, ma'am. Shall I go to the vote of thanks, ma'am?
Yes, madam. And reporting? Any
reporting? Yes, ma'am. Yes, ma'am. Sudha Devi ma'am will do the reporting. She is ready with the report.
Okay.
So can we go for the reporting?
Yes, madam.
Okay, okay. Sudha ma'am?
Yes,
ma'am,
ma'am.
unmute your mic. Yeah.
Yes, ma'am.
Devipan sir also is here. Devipan sir also is here.
Yes,
After
sir,
that,
yes,
Devipan
sir.
sir will.
Oh, yes, sir.
Shall I, ma'am?
Yes, ma'am. Sudha ma'am, you can start.
Thank you. I am privileged to present the report of Day 2 for the 2-day National E-Conference on Biodiversity Building and Natural Resource Management. The day started by invoking God Almighty by Ms. Yangchen Dolmiakom, research scholar, Department of Botany, Avinashlingam Institute for Home Science and Higher Education for Women, Coimbatore. Dr. Pambi Ghosh, Assistant Professor and Head, Department of Botany, Seva Bharati Mahavidyalaya, Jharkhand, West Bengal, the Secretary of the conference, welcomed the gathering. Professor J.P. Kashuri, Department of Botany, University of Burdwan, West Bengal, and Professor Srija Dutta, Controller of Examination, Vidyasagar University, West Bengal, felicitated the gathering. Introduction about the day 2 was given by Dr. V. Gayatri, Assistant Professor, Department of Botany, Avinashlingam Institute for Home Science and Higher Education for Women, Coimbatore. On the day 2, we had 4 invited talks. The first talk is by Dr. K.R. Mishra, Assistant Professor, Department of Wildlife and Biodiversity Conservation, MSCBD University, Odisha, on assessment of biophysical parameters and plant diversity of Silimpar, an ecological approach in conservation planning. The second talk was given by Professor Vidyanatja, retired principal, Professor of Botany, Ellen Mihal University, Bihar, on utilization of components of biodiversity in food plants of Mittal, North Bengal, Bihar. Sorry. The third talk was given by Dr. Nirmal Goho, Assistant Professor, Department of Environmental Science, Tripura University, Assam, on biodiversity under changing climate, mitigation of threats. The last invited talk was by Dr. D. Tangamani, scientist, FGRM Division, IFGTP, Coimbatore, Tamil Nadu, on conservation and characterization of forest genetic resources and its utilization for our future. After the invited talks, we had technical session 2, where 13 participants presented their paper. The session was chaired by Professor R. Bhattacharya, retired professor, Presidency University, Kolkata, and there were 4 subject experts, where 13 research scholars and faculty members presented their paper. The first paper was presented by Shreya Rao, PG Department of Botany, Lady Barbara College, Kolkata, West Bengal, on synergetic activity of tea with some common medicinal plants, a review. Then Maria D. Brahma from the Department of Forestry and Biodiversity, Tripura University, Tripura, presented a review paper on potential of freshwater macrophytes for phytoremediation of wastewater, followed by Bhuvaneshwari, PhD scholar, PG and Research Department of Botany, Vellalar College for Women, Erode, Tamil Nadu, presented a review on Ceropegia juicea. Then Ume Hassan, PG Department of Botany, Lady Barbara College, Kolkata, West Bengal, presented a review on influence of elevation on the caffeine content in tea and coffee. Then Srijita Shreel from the Department of Forestry, Uttara Benga Krish Vishwavidyalaya, West Bengal, presented a paper on reviving urban forestry for sustainable cityscapes, an insight to the refuge of existing natural resource. Gautam Ghosh, Department of Botany, Vijay Narayan Mahavidyalaya, West Bengal, presented a paper on role of plants bioresource in traditional healthcare system, a case study from the Upper Sigar district of Arunachal Pradesh, India. Dr. Rabinder Day, Assistant Professor in Chemistry, Kocho Bihar College, presented a paper on spices as natural products and their medicinal values. Angita Dutta, Center of Life Science, Vidyasagar University, presented a study of pollen diversity of vegetation of Minapol down during post-monsoon season. Deepu Samantha presented. Deepu Samantha from Department of Botany. Dr. Kanil Bhattacharya College, West Bengal presented a paper on plants for soil pollution control, a review. Mitudi, Associate Professor, Department of Botany, Gurudas College, West Bengal, presented a paper on variation in grain morphology among dwelling indigenous rice biodiversity. Saril from the Department of Botany, Avinashlingam Institute for Home Science and Higher Education for Women, Coimbatore, presented a paper on pharmacological, phytochemical analysis and free radical scavenging activity of calcium citrus leaves. Lavanya, Department of Botany, Avinashlingam Institute for Home Science and Higher Education for Women, Coimbatore, presented a survey on floral biodiversity in Avinashlingam Institute for Home Science and Higher Education for Women, Campus 1, Coimbatore, Tamil Nadu. In the last paper on green synthesis and characterization of silver nanoparticles from Lycopris species have been presented by Manjula Devi, MPhil scholar, Department of Botany, Avinashlingam Institute for Home Science and Higher Education for Women, Coimbatore, Tamil Nadu. Thank you, ma'am.
Thank you, Sudha ma'am. Now I would like to wind up the session with the vote of thanks. Every teacher once was a student. Every winner once was a loser. Every expert once was a beginner. But all of them have crossed the bridge called learning. As we express our gratitude, we must never forget that the highest appreciation is not to utter words, but to live by them. John F. Kennedy. So with this quote, I would like to propose the vote of thanks on the second day of the two-day National E-Conference on Biodiversity Building and Natural Resource Management, NCBBNRM 2021. I express my heartfelt thanks to Professor J.P. Keshri, Department of Botany, University of Burdwan, West Bengal, and Professor Siraj Datta, Controller of Examinations, Vidyasagar University, Midnapur, West Bengal, for their wonderful greetings. Thank you, professors. My sincere thanks to Dr. Pampi Ghosh Ma'am, Assistant Professor and Head, Department of Botany, Sheva Bharati Mahavidyalaya, Khapgari, and Secretary of the Conference for her welcome address. Thank you, ma'am. I owe my sincere gratitude to the invited speakers of the day, Dr. R.K. Mishra, Assistant Professor, Department of Wildlife and Biodiversity Conservation, MSCBD University, North Orissa, Professor Vedana Jha, Retired Principal and Professor of Botany, LN Mithila University, Bihar, Dr. Nirmali Gogai, Assistant Professor, Department of Environmental Science, Tezpur University, Assam, and Dr. D. Thangamani, Scientist, FGRM Division, IFGTB Coimbatore. Thank you, professors, for your excellent speech. Your thought-provoking lecture will help humankind to protect and manage the biological diversity available in the earth for a sustainable environment. My heartfelt thanks to all the paper presenters in both the technical session 2 who presented their research work as well as review work on various aspects of biodiversity conservation and natural resource management. Thank you all. I deem it my pleasure to thank the chairperson of the technical session, Professor R. Bhattacharya, Retired Professor, Presidency University, Kolkata, for their valuable time to conduct the session. Thank you, sir. It's my pleasure to thank the subject experts of technical session 2, Dr. A. Gayal Nivedita, Professor of Botany, Abba Sahib Garwar College, Pune, Dr. Vinita R. Bhatta, Assistant Professor, Department of Zoology and Genetics, Jyoti Nivas College, Bangalore, Dr. Somapal Saha, Associate Professor, Microbiology, Lady Brabourne College, Kolkata, Dr. Nadesan P. Nadimuthu, Assistant Professor, Botany, Tagore Government Arts and Science College, Puducherry. Thank you, professors. My deep sense of gratitude to the prayer song student, Ms. Yangchen Dolmakom, the research scholar from Department of Botany, Avinash Lingam Institute for Home Science and Higher Education for Women. Thank you, ma. It's my privilege to thank the rapporteur of the technical session 2, Dr. Pampi Ghosh, ma'am, HOD, Department of Botany, Sheva Bharati Mahavidyalaya, Khapkari, Jhargram, West Bengal. Thank you, ma'am. I'm very grateful to the reporter of the day, Dr. S. Sudha Devi, Assistant Professor, Department of Biochemistry, Biotechnology and Bioinformatics, Avinash Lingam Institute for Home Science and Higher Education for Women. Thank you, ma'am. My sincere thanks to the ICT professionals and non-teaching staffs for their timely help in making the necessary arrangements for today's program. Thank you one and all, and thank you for your patient listening for the whole session. Thank you. Das
So
sir?
thank you all. Dr. Das.
Yes.
So this
thank
is
you, madam, for your presence.
Thank you, sir.
Make it complete. And we will meet again together at the evening, 6:30 p.m. sharp.
Yes. So can
So
we have the
today's
Google Meet link alone?
Same.
Yes, sir. Meet link alone we can have.
Yes.
Okay, sir. Okay, sir. So dear participants, please join us tomorrow at 6:30 p.m. sharp so that you can hear to some more presenters on their research findings, and then you can complete your